kids, are we live? Hello. Hi everyone, am I live? So welcome to the session everyone. Welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma, and today we are going to continue the biomolecule part. So today we will revise protein, lipids, nucleic acids, right? This is what we are going to revise today. So I hope you people are excited. I hope you guys have revised everything. Okay. So in this chapter, I will not finish it, in 2 hours, 3 hours or 4 hours. Why? Because this chapter, I have seen that students, they generally face problem in this particular chapter. So we will understand all the aspects of this chapter slowly, and then we will finish it, and we are going to keep one MCQ revision session for the same as well. But that will be on your Plus platform. I will take a special class. We'll let you know about it. Fine, we'll let you know about it. So any other doubt from the last session? Anything that you people want to ask? Good evening. Good evening, good evening, good evening. And let me tell you one thing, students, I know very less number of students are, attending the session these days. If you're thinking that in last 6 months or in last 4 months, you will be active and then you will cover your syllabus and then, you will study day and night and you will be able to crack the NEET examination. I'm sorry, it is not the way. You have to be consistent, okay? You have to be consistent. And trust me, if you will attend this NEET Conquer Batch properly, right, your chances of selection will be higher then. Because we provide you the content, right, detailed content in these marathons, in these one shot, and that will help you out to clear the concept. Then you guys can revise it from the NCERT, and then you people can practice the MCQs as well. Okay, you guys can join our AIPMT as well for the MCQ practice, for the mock test. And yes, I have seen many students are commenting again and again, ma'am, Plant Kingdom Part 2 is not available. Don't worry, I will take another session of it. I don't know what happened, right? It is deleted basically. I don't know why. So we will take it again. Okay. But my point is, again and again, you all are asking Plant Kingdom Part 2, Plant Kingdom Part 2. You didn't check the cell cycle yet. You didn't check the animal kingdom yet. So why are you after that part only? Why? Yeah? Why? Done? Okay, so let's continue the biomolecules. Today we are going to start from the protein, then lipids, then the nucleic acid, and finally the enzymes as per the time. Okay, so before that, first of all, not this one of course. Just have a look. See, today at 2 p.m. I have posted something, and that is the life cycle of plant kingdom, right? So yesterday I started this, I posted one video and I told you that I'm going to start a short series, and in that short series what will we do? We will cover all the important and the tricky concepts of the biology. But to continue this series, I need your support. Let me tell you honestly, okay? If I will not get support, so obviously I'll end it. I will not be able to continue. So what you have to do, be regular, be consistent, check out this video, share it with your friends as well, bache, right? And whatever topic you want next, just put that in the comment section. And yes, of course, do let me know how was the recording as well, how was the session. Here you people can see, right? Here you guys can see. And see here, HSP sir has posted this, the books wala part. So you guys can follow our channel and you will get all the important updates. Okay, you people are going to get all the important updates. And moreover, bache, this is a strategy. It is a very good strategy video. And here in this video, I told you that how can you cover the syllabus. Okay, so do watch it. If you have checked it, very good. Otherwise, do watch this video. And moreover, if you want to join the batch or the mock test series, here you guys can see the description box. So what do you have to do? Just click here, right? And let me tell you, we are soon going to close the entries for this batch. Okay, so just join it. You know that Avenger coupon code is already applied and you will get this batch at a price of 14999. And from 1st of August, I'm going to start the plant physiology. So do not delay. That's a request. Okay, do not delay. Now, let's revise it quickly. So when we talk about the biomolecules, if I follow the NCERT, so as per NCERT, biomolecules, right, all the carbon containing compounds are biomolecules, isn't it? Isn't it? Right? All the carbon containing compounds are what? They are the biomolecules. That's what we have covered so far, isn't it? And I told you that when we talk about the biomolecules, we even discuss biomicromolecules. We even talk about the biomacromolecules, right? Biomicromolecules and the biomacromolecules. And when it comes to the biomicromolecules, the molecules having, they are basically obtained from the living organisms. They are basically obtained from the organic things. And when it comes to the biomicromolecules, so biomicromolecules, they are having the atomic weight, having the molecular weight rather, less than 1000 Dalton. Right? Right? Their molecular weights are, in the case of biomolecules, molecular weight is less than 1000 Dalton. And when you talk about the biomacromolecule, obviously the molecular weight is more than 10,000, here it is more than 1000 Dalton, here it is more than 10,000 Dalton. Okay, that comes under the biomacromolecule. So how did we start? We started with this part, with the chemical analysis. This is what we have covered already. Okay, so I'm just giving you the quick recap. Then we are going to start from the amino acid and from the proteins part. So you know that we have to take the living tissue, then grind that living tissue in trichloroacetic acid. Right, in trichloroacetic acid. And why trichloroacetic acid? Because it can precipitate your biomacromolecules. Right, it can precipitate your biomacromolecule. So we will get a thick slurry. So we have to strain it with the help of a cheesecloth. So after straining, we will get two fractions. One is the acid soluble part. Right, another is the acid insoluble part. So here we have a trick, bache, that is fast. F stands for filtrate. A stands for acid, S stands for soluble. So the filtrate is going to be acid soluble, isn't it? Filtrate is going to be acid soluble, and this filtrate will contain the biomicromolecule. This filtrate will contain the cytoplasmic composition without cell organelles. Right, without cell organelles. Clear, Subhashini? Now, when you are talking about the retentate, okay, so retentate will include what? It will include biomacromolecules having molecular weight more than 10,000 Dalton, more than 1000 Dalton. Clear, bache? This is what we have in the retentate. So in the biomicromolecule, you will see the examples of your monosaccharides, even the disaccharides. Right, you will see the examples of your amino acids, nucleosides. Okay. Okay, this is what we are going to see here in biomicromolecules. And when they are the biomacromolecules, we have the examples of polysaccharides, we have the examples of proteins, the nucleic acid, like this. So this is what we have covered so far. Okay, okay. And this retentate also represent the cytoplasmic composition with that organelle part. Okay, it is without organelle composition, it is with organelle composition. So that is what we have covered so far. So you guys can check the last part as well. Okay, so after this we revised the monosaccharides, right? The single sugars they are. So now you have to do one thing. You have to tell me that which bond you are going to see in the case of carbohydrate. Two monosaccharides, they are joined with the help of which bond? Tell me quickly. Two monosaccharides, they are joined with the help of which bond? Quick. Yes, Ishwat, Siva, Akanksha. Exactly. That is going to be the glycosidic bond. Okay. That is going to be the glycosidic bond, isn't it? Isn't it? So glycosidic bond is the one which will connect two sugars together. Okay, so here you guys can see when they are the monosaccharides, right, bache, and the carbon number can vary from 3 to 7. So it can be triose, tetroses, pentoses, hexoses, and heptoses, right, as per the carbon atom. And here we have the examples, and we have covered it in detail about the aldoses, about the ketoses. So do check that video, bache, do check that last part. So today we are going to start from the proteins. Okay, what we are going to discuss today, we are going to talk about the proteins. Fine, just a minute. Yes, so we are going to talk about the proteins. So proteins, they are present in acid soluble fraction or in acid insoluble fraction? Tell me, proteins, where are they present? Are they present in acid soluble fraction or in acid insoluble fraction? Where are they present? Be quick. Proteins. So proteins are also the bio-macromolecule. Okay, proteins are bio-macromolecule, and these proteins are the part of acid insoluble fraction. Okay, it is the part of acid insoluble fraction. It means its molecular weight is more. Now when you talk about the proteins, as I said, it is right, as I said that it is what? Yes, it is what? It is a polymer. What is it? It is a polymer, Akanksha. So if it is a polymer, then of course it is made up of some monomers. So when you talk about the proteins, the monomers that we are going to discuss is amino acids. So firstly, we will talk about the amino acids, and then we are going to discuss the proteins. Clear? Then we will discuss what? Then we will discuss the proteins. Just a minute, I need my colors. Fine. Fine. Then we are going to talk about what? We are going to talk about the proteins. So now when you talk about amino acid, what is the amino acid? What is the amino acid? Amino acid, bache, amino acids in nature, they are, you can say that 200 to more than 200 to 300 amino acids are present, right? But when it comes to the protein making amino acids, we have only 20 amino acids. Clear, bache? Clear? If you talk about the existence of amino acids in nature, so they are more than 200 to 300, but when it comes to the protein formation, we talk about the 20 amino acids. So when you talk about these amino acids, they are the part of your acid soluble fraction. So obviously they are biomicromolecules, isn't it? They are biomicromolecules, isn't it, bache? So they are the monomers which are going to form the protein. Clear? These are the monomers which are going to form the proteins. So now, yes. So let's continue it from this part. So you all can see this diagram, isn't it? This is the basic structure of an amino acid, right? Again, we are going to complete this part in the, we are going to complete the biomolecule in the parts, bache. Right, we will divide the chapter into different parts, and then we will discuss each and every part in detail, because this chapter is important. Okay, biochemistry is very important in your MBBS as well. Okay, so don't be like that I need to crack the NEET examination, so I just need to, you know, finish this chapter. No, try to understand it. It's a very interesting chapter. It is actually it's a very interesting chapter, and you will love it, you will enjoy it. Okay, after this biomolecule Balmiki, we will go for the structural organization in animals. Don't worry about that. Okay, after this chapter, we will start the structural organization. So now, what are we going to start, bache? We are going to start the basic structure of amino acids. What do we need to start? We need to start the basic structure of amino acid. So here you guys can see the diagram as well. Let me draw this. So here you will see we have the carbon. Now when you talk about the carbon, and yes, I'm repeating this line again. As per NCERT, all the carbon containing compounds are what? They are the biomolecules. Right? They are the biomolecules. Bache, these lectures are more than sufficient for NEET. Nikhil? Okay. Right? So now in the case of carbon, we know that carbon is tetravalent. Right? It is tetravalent, having 4 valence electrons, isn't it? And you know that carbon is having the property of the catenation. But here we don't need catenation, we need something else. So you know that carbon is tetravalent. Okay, carbon is what? Carbon is tetravalent. Now, you know the structure of methane, that is CH4. So when you talk about the amino acids, we used to call them as substituted methanes. Do you know that? We call them as substituted methanes. So carbon is having 4 bonds. So on one side you will see the amino group, that is your NH2. On another side you will see the carboxylic acid, COOH. On another side you will see the hydrogen, clear, the hydrogen, and here you will see R. This R is what? This is the alkyl group. What is it? It is alkyl group. And here you can even see R, you can even see H, that is hydrogen. So this is the basic structure of an amino acid. What is it? It is the basic structure of an amino acid. So carbon is having this amino group, carbon is having this carboxylic group, it is having this hydrogen, and here you can get the alkyl group as well as the hydrogen. Alkyl group as well as the hydrogen. Now, this is the basic structure. So all the amino acids, in all different type of amino acids, it is the R group which will change, and on the basis of that they will have different properties. Clear bache? They will have different properties. So as I said, more than 200 to 300 amino acids are there in nature, but when it comes to the protein forming amino acid, right, when it comes to the protein forming amino acid, so we have 20 amino acids. What do we have? We have 20 amino acids. Now actually the number is 22. We consider 2 extra amino acids as well, but in your NCERT only 20 is given, so we will discuss as per that. Okay, so this is the basic structure as I said. Now what you need to compare, let me tell you. See, so now let's say instead of R, here we have the H, here also H, NH2, and this one is COH. So bache, it is your simplest amino acid, that is glycine. Okay, it is the structure of your simplest amino acid, right? It is the structure of your simplest amino acid, which is glycine. You can see this. So first of all, you should know the basic structure of amino acids. Then at the place of R, it should be H. So if it is H, so it is the glycine. Okay, it is the glycine. Now, when you look at this particular structure, instead of H, if we have R here, so can I say that this carbon is attached to 4 different groups? Yes. Can I say that this carbon is attached to 4 different groups? So we also call it as alpha carbon. Right, this carbon is what? This carbon is asymmetrical, isn't it? This carbon is what? It is asymmetrical. It is attached to 4. It is attached to 4 different groups, isn't it? It is attached to 4 different groups. We call it as alpha carbon, bache. It is asymmetrical carbon. So for amino acids, we also call them as alpha amino acids. Right? For amino acids, you can also call them as alpha amino acid, leaving glycine, right? So glycine is an exception here. Why? Because in the case of glycine, this carbon is attached to the hydrogen here. It is attached to the hydrogen here. So glycine is an exception. Glycine mein carbon is not asymmetrical. It is not alpha amino acid. Otherwise, all other amino acids are what? They are alpha amino acids. So any doubt here? Yes, everyone. Do you have any doubt here? Do you have any doubt? Yes? Done? Done? So this is the glycine. So whenever we will change the R, the functional group, accordingly we will get different type of amino acids. So now let's revise the amino acids quickly. So do you know about these 20 amino acids? Do you remember the name? You know, this part is also related with your class 12th genetic code. In the genetic code, we used to draw a box. We talk about the different types of amino acids there. Some codes are given, and this triplet base codon is going to form one amino acid. Right? So sometimes in the paper, they used to ask you that particular question. So obviously you need to master that too if you are targeting to get 360 out of 360. If your target is 330 plus, like in between 330 to 350, then you can ignore that. Otherwise, you should revise it. Okay? You should revise it. So these are the basics. So now, see, methane is CH4. Methane is CH4. So here, can we say that these amino acids are substituted methanes? The hydrogen is replaced by amino group. Hydrogen is replaced by carboxylic acid group. It is replaced by this alkyl group, isn't it? So obviously it is justified that these are the substituted methanes. These are the substituted methanes. Now, let's talk about the names of these 20 amino acids, because we have so many examples here in NCERT. Okay, so I believe that you people should, remember this, right? So I'll give you, you can take it as a challenge. You have to remember the name of amino acids in less than 1 minute. Can you do it? You have to remember the name of all the 20 amino acids in less than 1 minute. Can you do it? Chalo, theek hai, I'll give you 5 minutes. Can you do it? I think yes, you all can, and you should give it a try. Okay, let me start. Even I remember that. So, alanine, arginine, asparagine, okay, aspartic acid, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, proline, that's phenylalanine, serine, tyrosine, threonine, tryptophan, and the valine. So tell me, do you remember it? Yes. Yes. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Okay. So I know all the, I know even the structure of these amino acids, right? But so you can remember them in an alphabetical order. Okay. You guys can remember it in an alphabetical order. So we start with alanine. Alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, proline, phenylalanine, serine, tyrosine, tryptophan, threonine, and then comes the valine. Okay, this is what we have. These are the name of the 20 amino acids. So I'll give you 5 minutes to memorize it. Okay, even after the class, I'll give you 5 minutes. Okay, I'll give you 5 minutes. Okay, so now when you look at it, obviously, see, cysteine, sorry, not cysteine, glycine. Yes, glycine. It is the simplest amino acid, right? It is the simplest amino acid, as I said. So when you talk about these amino acids, you have seen the basic structure. Now you can say that these amino acids contain carbon, they contain oxygen, they contain hydrogen, nitrogen, and sulfur as well, right? And sulfur as well. So now this is what we need to discuss. So as I said, what do we have? We have 20 amino acids. Two more are added. One, that is selenocysteine and pyrrolysine. Okay, I told you now that they are basically 22 now. So selenocysteine and pyrrolysine. Selenocysteine and pyrrolysine. Clear, bache? Clear, bache? These are the 2 extra amino acids. Clear? And we have some non-protein forming amino acids also, like your citrulline, ornithine. I hope you know about it. Okay, I hope you know about it. So now, as I said, here in the amino acids, bache, we will see the basic amino acids. We will discuss the acidic amino acid. We will discuss the aromatic amino acids. We are going to discuss the neutral amino acids. We will discuss the essential amino acids. So this is what we need to cover here in these amino acids. Clear, bache? This is what we need to cover here. Okay. So now when I talk about the properties of these amino acids, so do you know that their structure, it varies in different solutions? Yes. Do you know that? Do you know that when you talk about the amino acids, their structure, it changes in different solutions, right? Right? Tell me, their structure, even it gets changed in different solutions. Are you aware of it? Huh? Are you aware of it? So obviously they have the amino group, they have the carboxylic group. This NH2 can make NH3 positive, the COH can make CO negative. So obviously they have ionizable nature, they can form the ions, isn't it? They can form the ions. So that is why in the solutions of different pHs, right, you will see that these amino acids will give different type of structure. So here we can say that they can form the cation, positively charged, anions as well, and negatively charged. Clear? Cations as well, anions as well. Now related to it, we need to cover one topic, and that topic is of your Zwitter ion. Do you know about it? Yes. I think I didn't add the structure here, but that's fine. Okay, so what we need to discuss here, we need to discuss the, yes, we need to discuss the Zwitter ion here. Fine, Zwitter ion here. So as I said, the amino acid is what? Amino acid. As I said, amino acid, they are amphoteric in nature. Do you know the meaning of being amphoteric? Yes, bache, they have both the acidic carboxylic group as well, the basic NH2 group as well. The acidic carboxylic group as well, the basic NH2 group as well. And as I said, amino acid is amphoteric in nature. Now, just look at the one thing. See, when it is like this, NH3 positive, here let's say it's R, H, and it's CO. Yes, it's CO positive, COH. Now, just have a look. Okay, so now what is the Zwitter ion? I'm going to explain you Zwitter ion, which is also known as dipolar ion here. But this is the important part, okay? Even you will discuss it in the chemistry. Now see, this, because of the presence of amino group, as I said, it can form cation. Because of the presence of this carboxylic group, as I said, it can form anion as well, right? It is going to form the anion as well. Now this Zwitter ion, see, it's dipolar. Can you see it? It's dipolar, right? So COH, this carboxylic acid, it will give its hydrogen to this amino group. It will become NH3 positive, and this one will become CO negative. So what is it? It is a Zwitter ion. It is dipolar. Plus and minus both are present. So one plus charge, one minus charge. So that is why it is even the electrically neutral. So net charge is 0 na. So it is electrically neutral. Is that clear? Yes. Is that clear? So it is showing that amphoteric nature in the case of these amino acids. Right, bache? So due to the presence of this plus and minus charge, the amino acids, they are considered as what? They are considered as the Zwitter ions. Any doubt here? Yes, bacho. Any doubt here? So as I said, they are substituted methanes. They are going to form the Zwitter ions. Right, bache? Total 20 amino acids are there. Right, total 20 amino acids are there. So all these points are important, and you will get a question. Okay, you will get a question on the basis of it. Yes, so it is electrically neutral, having two charges, one plus, one minus. So plus minus will be equals to 0. Okay, plus minus will be equals to 0. So this is the Zwitter ion. Now, as I said that you should remember these 20 names. Why? Because bache, see, we have to complete different examples. Okay, like let me start. Just in the paper, they can ask you name basic amino acids. Okay, name basic amino acids. So for basic amino acids, the trick that we have is HAL. H-A-L. So H stands for histidine, A stands for arginine, right? And L stands for lysine. So these three are the example of what? They are the example of basic amino acids, right? Right. And in that, yes, and in that list, if you have seen it carefully, even acidic amino acids are given there, isn't it? even acidic amino acids are given there. So when it comes to the acidic amino acid, you have two things, two examples. One is aspartic acid, another is your glutamic acid. Clear, bache? One is your aspartic acid, another is your glutamic acid. The another question that will come in the paper is sulfur containing amino acids. Right? I told you now that in amino acid, sulfur is also present. So sulfur containing amino acid. So it includes, the trick here is CMS. What is the trick here? It's CMS, Chief Minister, sir. What is the trick here? Let me write it here. Chief Minister, sir. So Chief Minister, C stands for cysteine. C stands for cysteine. M stands for methionine. Okay, and S stands for sulfur. So CMS means sulfur containing amino acid, that is your cysteine, that is your methionine. Right, bache? That is your cysteine, that is your methionine. So any doubt here? Yes, bacho. Any doubt here? Sure? Sure. So now how do we say that this amino acid is a basic amino acid? On what basis we used to differentiate it? This is the question here, that on what basis we used to differentiate that, okay, this one is the basic amino acid, this one is an acidic amino acid. On what basis? Obviously on the basis of presence of groups. So when I talk about the basic amino acids, they have extra amino group. Right? What will you see there? Extra NH2 group here. Like here in histidine, arginine, lysine, you will see instead of 1, you will see 2 NH2 group. Right? What are you going to see here, bache? You people will see that there will be 2 NH2 groups. Okay, there will be 2 NH2 groups. So 1 carboxylic group and 2 NH2 groups. Fine, 2 NH2 groups. Clear, bache? Now it is the acidic amino acid. Here, 2 carboxylic group and 1 amino group. Clear, bache? Clear? So when it is sulfur containing, obviously sulfur is present. And when do we say that the amino acids are neutral? When do we say that amino acids, they are neutral? When they have one NH2 group and one COOH group. Okay, one NH2 group and one COOH group. Right? Right? So here you can see, leaving these five amino acids, rest 15, they are what? They are the neutral amino acids. Clear? They are the neutral amino acids. Is that clear? Yes, bache, Priyanka. So acidic, basic amino acid, whole, acidic amino acid, and then the sulfur containing amino acids. Clear, bache? Then the sulfur containing amino acids. So any doubt here? Yes, quickly tell me. And then the next question that we have is the aromatic amino acids. So do you know the example of aromatic amino acids? Yes. Do you know the example of aromatic amino acids? Yes, bache, Manish, we'll talk about it. The next example that you need to remember is what? It is the aromatic amino acid. Now, what is the meaning of aromatic amino acid? Aromatic, the ring is present there, isn't it? The ring is present there. So it's T2P. Okay, what is it? It's T2P, right? What is it? It is T2P. It is a previous year question. Fine, it's a previous year question. So T2P means you have tyrosine. Yes, you have tyrosine. You have tryptophan. And P stands for phenylalanine. Okay, P stands for phenylalanine. Is that clear? So tyrosine, tryptophan, phenylalanine. Tyrosine, tryptophan, phenylalanine. So aromatic amino acids, it's T2P, tyrosine, tryptophan, phenylalanine. Clear? Clear? So we have basic amino acid, acidic amino acids, sulfur containing amino acid, neutral amino acids, and the aromatic amino acids. So as per NCERT, this is what you need to remember. Clear? This is what you need to remember. Fine? Fine, is that clear? Is that clear? Let me show you some Diagram. So here, just have a look, bache. This diagram is from my favorite book. It is the favorite book of biochemistry, Stryer. I really like it. Yeah. This one is from Leninger, I guess. So, but my personal favorite book is Stryer Biochemistry. Right? You will get the chance to study it in your MBBS. So crack your NEET examination as soon as possible. Then in the MBBS, just pick up your biochemistry. I know many of you hate biochemistry, but I really love it. Okay, so my favorite is Stryer. That's a very, very good book. And very famous book for biochemistry is also the Leninger. But I always prefer Stryer, love it. Okay, so here you guys can see 21 proteinogenic alpha amino acids, right? And I told you now it is 22. So here you can see amino acid with electrically charged, so positive one. So positive one means your basic amino acids, right? So basic amino acids are what? They are positively charged. Of course, it is basic. So acidic amino acid means it is negatively charged. Clear, bache? It is negatively charged. So when it is positive, the trick is all right. Histidine, arginine, and lysine. And here you guys can see that here you people can see 2 amino groups will be there. Fine, 2 amino groups will be there. Right, bache? Right? Okay. And when it is aspartic acid and glutamic acid, 2 carboxylic groups will be there. Fine, 2 carboxylic group will be there. Yeah, I will share the PDF in the Telegram group after the completion of chapter. So see, these are the amino acid with polar uncharged side chains. Like when you talk about the side chains, you're talking about that R group. So sometimes you have uncharged, they are polar, but they are uncharged side chains as well, right? Special cases are also there. So obviously whatever I have marked as important, this is what you need to cover. That's all. It is just for your knowledge, but whatever I have marked as important, that is what you need to cover. Clear, bache? That is what you need to cover. So here you guys can see histidine, arginine, and lysine. So histidine, right, NH3. In the case of histidine, like in some books, even it is given that in some books, they only consider arginine and lysine as the basic amino acid, right? They didn't mention the histidine there. They didn't mention the histidine there because histidine in different pH, it behaved differently. But overall, your histidine, arginine, lysine, this is what you need to consider when you talk about the positively charged or the basic amino acids. Any doubt here? Sorted? Clear? So here you guys can see histidine having these amino groups, and now this arginine, clearly you can see NH3, and this is the lysine, clearly you can see NH3. See, rest everything is same. This side amino group, this side your carboxylic group, here you have hydrogen, and this one is the alkyl group. So in lysine, 1, 2, 3, then amino group. So ultimately CH2 amino group. Here you can see CH2, 1 NH, and then again to this carbon, that is how it is attached. Right, bache? And here you have the histidine. Now next is your acidic amino acid, aspartic acid, and glutamic acid. Right, bache? So 1 carboxylic group here, 1 carboxylic group here, 1 carboxylic group here, 1 carboxylic group here. So just see the difference. It is a previous year question. Okay, it is a previous year question. Just see the difference here. In the case of aspartic acid, carbon, amino group, carbon, carboxylic group, hydrogen. So when you talk about the side chain, CH2 and then COOH, here 2 CH2 are present, right? 2 CH2 are present, that's all. Rest structure is all same. Okay. So this is about the aspartic acid and the glutamic acid. So bache, aspartic acid, right? When it is COOH, you will say aspartic acid. If it is COO negative, then we use the word aspartate. Remember, aspartate means ionic form. If glutamate is written, right, don't get confused. It means ionic form. Just aspartic acid means ions are not formed yet, it is COOH. If it is aspartate, it means aspartic acid has formed that ion, right? Glutamic acid ka glutamate. So in detail your HSP sir can explain it in a better way, but this is what you need to remember. See, this question is the previous year question, so I'll recommend, yes, here you guys should focus on the structure. And moreover, alanine, serine, and glycine, the structure of these 3 amino acid is given in your NCERT, so you should remember that too. So whatever is given in NCERT, that we cannot leave. Fine, that we cannot leave. And this one is a previous year question, not the recent one, but again it's a previous year question. So according to me, you people should know the structure. And yes, bache, histones are DNA, it gets coiled over these histones, right? The DNA, it will coil over the histones and it is going to form that structure. Remember, nucleosome. So these histones are what? They are positively charged amino acids. They are positively charged proteins. Why? Because this histone is made up of basic amino acid. Okay, it is made up of basic amino acid. Lysine is more here. Lysine, arginine, both are present, but lysine is more here, right? Done? So it is important. It is again a previous year question. Clear, bache? Clear, bache? So now quickly tell me the example of basic amino acid: histidine, arginine, lysine. Example of acidic amino acid: aspartic acid, glutamic acid. Example of your aromatic amino acid: T2P, your tyrosine, your tryptophan, and then comes the phenylalanine. Your sulfur containing amino acid, the trick is CMS, Chief Minister Sir, cysteine, methionine, they are two sulfur containing amino acid. So rest 15 other than these positively and negatively charged amino acid, they are the neutral amino acid, like your valine is electrically neutral. Clear? Your valine is electrically neutral. Clear, bache? Clear, bache? So here again you can see the diagrams. Now bache, I will add one more thing here. See, these amino acids are also having the symbols. Jaise in the class 12th, in the genetic code, we used to write it like this. It's very common. TRP. It means tryptophan. है ना? This is how we write them. है ना? है ना? TRP, tryptophan. This is your phenylalanine, right? So these are the short forms. But other than short forms, they are also having the symbols also, right? They have the symbols also. Like here you can see arginine. We are calling it as R. Arginine. What is it? Arginine. R is given. Histidine. H. Lysine. K. So lysine is not L, it's K. So if L is given, it is leucine. It's not lysine. Your lysine is denoted by K. So these amino acids, they are denoted by these alphabets. Okay, and we also use the short forms for them. Just TRP, tryptophan, right, bache? ASP or aspartic acid, right? But it is aspartic acid, symbol is D. Glutamic acid, GLU, right? So this is a very common mistake. GLU means glutamic acid. GLU means glutamic acid. GLN means glutamin. It is glutamin. Fine, it's glutamin. Fine. Fine. So serine S, threonine T, asparagine N, glutamin Q, right? So glycine G, proline P, cysteine C, selenocysteine U, alanine A, valine V, isoleucine I, leucine L, methionine N, phenylalanine F, tyrosine Y, and tryptophan is a W. Okay, just say your tungsten is W. Tryptophan is also W. Okay, so I'll recommend one thing here, bache. See, you can just mark this arginine, you can mark asparagine, glutamin, lysine, aspartic acid, glutamic acid, like those amino acid which are not represented, right, which are not denoted with the first alphabet. Okay, with the first alphabet, or sorry, with the first letter of their name, right, that you can revise, that you can memorize. Clear, bache? That is the smart work. Just say I know your glutamin. Okay, this glutamin, it is denoted by Q. Okay, so your lysine denoted by K. So I remember it like this. Fine. So you can focus on that letters, on that amino acids which are not denoted by the first letter of their name. That's all. So I will share this PDF in the Telegram group so that you will get the reference. But ultimately what you need to revise, what you need to remember, this is what you need to remember. Okay, this is what you need to remember. Clear, bache? Clear? So now I'll add one more thing here that is important as well, of course. So do you know tryptophan? Tryptophan. This tryptophan is even important for the auxin formation. Do you know auxin? It is the plant hormone. So for the formation of auxin, tryptophan is required. And even, you know, now our mothers used to say that before sleeping we should drink the milk. Do you know about it or not? that before sleeping we shouldn't drink the milk. Why? Because in that milk tryptophan is there. So that tryptophan will help in the formation of serotonin as well, which is going to induce the sleep. Okay, which is going to induce the sleep. Fine. So there is a science behind. Fine, there is a science behind. So this is a very common question, but it is a previous year question again, that which amino acid is used for the synthesis of tryptophan, synthesis for auxin, which is a plant growth regulator. That is the tryptophan. Clear? Denoted by W. That is the tryptophan. Clear? Denoted by W. Okay, bache? Sure. So tyrosin, this tyrosin, it helps in the formation of skin pigment melanin. For the formation of the skin pigment melanin, we need tyrosin. Okay, tyrosin is required for the formation of skin pigment melanin. So these amino acids are having different roles. So if there is any doubt, do let me know, bacho. Yes, do let me know. Clear? Sure? Sure. So it is the glycine, which is the simplest amino acid, and it is your tryptophan, which is the complex amino acid. So if in the paper they'll ask you simplest amino acid, the answer is glycine. But which one is the complex amino acid? That is your tryptophan. That is your tryptophan. So this is about the amino acid. So I'm repeating it again, and I want you people to remember the name of these amino acids. So alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, proline, phenylalanine, PQR is serine, tyrosin, threonine, tryptophan, and you have the valine as well. Okay, proline, phenylalanine. That's all. Clear, bache? Clear, bache? So this is what you need to remember. So that's your task. That's your task. Fine. So the another thing that you need to remember is your essential amino acid. What is it? It is your essential amino acid. So now what is the meaning of essential amino acid? Again, it's very simple. But they should be the part of our diet because they are not synthesized by our body. Okay, they should be there in our diet because our body is unable to synthesize them. Essential amino acids. Right? Yes, but your voice, you can ask the question. Okay, done. You can ask the question. So essential amino acid, as I said, they should be in our diet. As they are not synthesized by body. Clear? So for this, and yes, even it is important. Clear, bache? It is important. So here the trick is private Tim Hall. There are different tricks, bache, right? So what you need to remember, it's private Tim Hall. It's private Tim Hall. See, Hall, L. So L here means leucine. L here means lysine as well. Clear? L here means leucine. L here means lysine as well. A stands for your arginine. A stands for your arginine. H stands for your histidine. Right? It stands for your histidine. Clear, bache? So P stands for phenylalanine here. Okay? Phenylalanine. V, you know that single name, that is going to be valine. I, you know that it is again a single name, that is isoleucine. M again is very easy, that is your methionine. Okay? That is your methionine. Now you have T. You have 2T. So tyrosin will not be there. Right? See, how can you remember this? Because tyrosin is required for the synthesis of our skin pigment melanin. Right? It is important for the synthesis of, right? It is important for the synthesis of melanin. So obviously it will be synthesized in our body. So we are left with what? We have threonine as well. And we have the, yes, we have the threonine as well. And we have the tryptophan. Fine. We have the tryptophan. Clear, bache? So these are your essential amino acids. So 1, 2, 3, 4, 5, 6, 7 and 9, 10. Now you must be thinking that, ma'am, here only 9 amino acids are given and you are telling us about the 10 amino acids. Basically, there are 10 amino acids which are the essential amino acids. And here you all can write one note that when it comes to the histidine and arginine, they are considered as semi-essential. Do you know that? These histidine and arginine, Okay, histidine and arginine, they are considered as semi-essential because in the case of adults, they will start their synthesis but not in sufficient amount. Clear, bache? So histidine and arginine, they are also considered as semi-essential because in the adult, they'll start their formation. The amount, the quantity is not sufficient, but in the childhood, this histidine and arginine should be the part of our diet. Okay, it should be the part of our diet. Right? It should be the part of our diet. Is that clear? So this part is also important. So I have added these screenshots. You guys can check it later after the class. Okay. And after it, we need to start the protein. So here you can see glycine, G, right? G. So R group is basically, you know that it is simplest, so H will be there. So in alanine, it is the part of your NCERT. CH3 is given. Right? R group is your CH3. In alanine, serine is also given. And you can also check valine. CH3 to CH. Right? So I will share the PDF in your Telegram group and you all can check it. Fine, bache? You all can check it. Done? So any doubt? Array. Yes. Any doubt? Okay, it is not working properly. Allow me 1 or 2 minutes. Done? Where is that slide? Yes, here we have. Fine. So here you can see glycine, H is there. CH3, CH2OH. These structures are important, right? Why? Because they are given in NCERT. Done? They are given in NCERT. Clear, bache? Okay. So now we need to start the proteins, but before that, you need to know what is the meaning of a dipeptide. Okay. What is the meaning of a dipeptide? And then I will start the proteins. Chalo, I'll start the protein first and then I will explain it. Now, when you talk about the proteins, now you know that proteins are very important, right? They are going to form that structure here in our body, like structural framework and all, right? We know that we need the proteins as well, right? Bache? So when you talk about the proteins, no doubt it is present in your acid insoluble fraction. Okay, it is present in your acid insoluble fraction and it is a biomacromolecule. Okay, the molecular weight is more here and these proteins, they are the heteropolymer of amino acids. What is the word here? The word is heteropolymer. What is the word here, bache? The word here is heteropolymer. Heteropolymer. Hetero means different, isn't it? Like in the case of carbohydrates, we were saying that we have the two options, homopolysaccharides and the heteropolysaccharides. This is what we were discussing in the carbohydrate. Homopolysaccharide, heteropolysaccharide, homopolysaccharide, heteropolysaccharide. But when you talk about the proteins, simply this is what you need to remember, that they are heteropolymers of amino acid. They are not made up of same type of amino acid. They will always have different type of amino acids. Okay, so they are the different, they are the heteropolymers, isn't it? They are the heteropolymers, isn't it? Yes. Any doubt here, bache? Any doubt here? Any doubt here? Sure. Sure. So these proteins, they are also known as polypeptides. I will explain why. They are also known as polypeptides. What are they, bache? They are also known as polypeptides. Clear? They are also known as polypeptides. I'll explain why. So you know that in our body, proteins, they are playing the very important role, right? We have seen their composition as well. You know, in our body, they used to play the different roles, right? Like, I'll consider the simple example. When you talk about the cell membrane, in the cell membrane, these proteins are present and they act like the channels, right? They act like the channels, isn't it? Isn't it? Or you can say that they act like the carrier to carry the things, right? So these proteins, their receptors are present on cell membrane. So they are also helping in the recognition. isn't it? It is also helping in the recognition, isn't it? So your enzymes are mostly proteins. Enzymes which are biocatalysts, which are going to catalyze the living reactions in our body, they are what? They are also protein. So most of the enzymes, they are the proteins. So see, that is how it is working. And even to fight with the infection, you know, immunoglobulins, antibodies. Do you know about it? Immunoglobulins, we used to call them as IGs, IgA, IgM. So immunoglobulins are also what? They are also the proteins. Fine. So these proteins are even helpful in maintaining the osmolarity in our body. Albumin. These proteins are also helpful in the formation of in the blood clotting, right? Fibrin. Remember fibrin. So ultimately proteins are playing a major role in our body, right? So what are they? They are the heteropolymer of amino acid. What are they? They are the heteropolymer of amino acid. So if we are talking about the protein, so obviously it is for sure that we need to join the amino acids. We need to join the amino acids, isn't it? Like in the case of carbohydrate, the bond was glycosidic. So here the bond that we will discuss is your peptide bond. Which bond is there, bache? We have the peptide bond. We have the peptide bond. Clear? Clear? So here you guys can see the carrier. So different roles of protein. You know that proteins are even acting like the hormones as well. Okay? So we will discuss it one by one. So as I said, these amino acids, they need to join. So in the proteins, what will you see? You will see the peptide bond. That is why we even call them as polypeptide. Now, whenever you write the amino acid, this is how we used to write. Okay. You can consider any example as of now. You can consider any example as of now. Like, that's how I'm going to write the proteins. That's how I'm going to write the proteins. So when you talk about this amino group, right? This amino group, it is mostly on the left side of the chain. Okay, it is on the left side of the chain. Fine. So proteins are basically the long chains of amino acid. So whenever you write the amino acid, on the left side we will write what? We are going to write the amino group. So this side of the chain will be the N terminal, and the another side where your carbon, right, where your carboxylic group will be there, that will be your C terminal, right? That will be your C terminal. This is what you need to focus. Okay, so here we have amino terminal, and the right side we will get what? We are going to get the carbon terminal. Okay, bache. So such amino acids are present in our body, and when amino acids, their structure is like this, so we call them as L amino acid, right? These are the L amino acid. What type of amino acids are they? They are the L amino acids. Any doubt? Yes, any doubt? They are the L amino acid. Clear? And when you talk about, one student was talking about the bacteria. Okay, so in the case of bacteria may, you know that the cell wall contains peptidoglycans. Okay, so peptidoglycans means proteins plus carbohydrates, right? Proteins plus carbohydrates. So mostly in bacteria you used to get D amino acid, but in our body you will get L amino acid. And that is why when you take the antibiotics, right? So somewhere that is how the antibiotics help. So that we will discuss in one short video, but as of now, this is what you need to remember. Okay, bache? So now, as I said, we need to join these amino acids if we want to make the long chain of proteins. So that is how we are going to write it. Okay, that is how we are going to write it. So ultimately, bache, it is nothing. It is the dehydration which will take place. Fine. It is nothing. It is just the dehydration that will take place. Let me write it properly. Like, see, here you have your N, and here you have your amino group, right? Hydrogen group. Let's say here R1 is the alkyl group, and this side will have C double bond OH, right? Now you have your next amino acid. So next amino acid will be clear? This is the next amino acid. Now what are you going to do here? You are going to release the water. Okay, you are going to release the water. Ultimately, it is also the result of dehydration. What is going to happen here, bache? dehydration once again. So there will be the loss of water when we form the peptide bond, right? So I'm going to write these two amino acids like this. See, you can even check the bonding. If there is any confusion, you can ask me. It is like this, yes or no? Yes or no? Right? So water will go out. So what you are left with? CONH. CONH. So this is what you people can see here. Okay, this is what you guys can see here. CONH it is. What is it? It is CONH. Done, bache? Fine. Fine. This is the peptide bond. Right, this is the peptide bond formed by the loss of water molecule. So this is how your amino acids, they will join together, bache. So obviously next amino acid, next amino acid, next amino acid, and then together they are going to make the long structures, right? So that is why we are saying that proteins are polypeptides. Poly means many, peptide bonds. So many peptide bonds will be there. Right, and these chains are polypeptides, right? These chains are polypeptides. So the formation of peptide bond is very important. So peptide bond ke liye we also call it like this, na, C double bond ONH. C double bond ONH. It is the peptide bond. C double bond ONH. Clear? Yes, everyone. Is it clear? Is it clear? Yes or no? Soumyadeep, we have already covered it in last class. Kindly check that. Okay. So C double bond ONH, peptide bond. Right. So, bache, when two amino acids are connected together, right? When only two amino acids are joined together, they are connected together, you are going to use the word dipeptide. Right? You are going to use the word dipeptide. Clear? You are going to use the word dipeptide. Done? Sure? Deepa, what do you want me to answer? Fine. So this is the dipeptide. Two amino acids linked together by a one peptide bond. So C peptide bond, it's CONH. CONH. So this one is your first amino acid. This one is your second amino acid. Okay, so C carboxylic group of first amino acid, amino group of second amino acid, they will form the peptide bond. Done, bacho? They will form the peptide bond. Done. So now we will talk about the structure here, of course. Okay. Yes. So we are going to talk about the structure here. Fine. So these amino acids, as I said, they will form the protein. So how? So what are we going to start? We are going to start the. We are going to discuss the structure of proteins. So, bache, in the structure, we will talk about the primary, the secondary, the tertiary, and quaternary structure. Okay, we are going to discuss the primary structure of the protein, the secondary structure of the protein, the tertiary structure of the protein, and the amino and the quaternary structure. Amino acid, like Deepa, what do you want me to answer? First of all, type the question properly. Right. So Soumyadeep, this L and D, it depends upon how we are writing NH2 and COH group. Okay, which one is L, which one is D? It depends upon the how the NH2 and CO your carboxylic group is placed. Clear, bache? So structure. So ultimately the linear amino acids. See, it is the position of amino acid even which is going to decide the properties as well. Right? Ultimately these amino acids are going to decide the properties of the protein, and how are they placed, it is going to decide that. So see, when there is the linear chain, one amino acid, second, third one, fourth one, fifth one, sixth one, like this. As I said, how are they connected, bache? How are they connected? They are connected with the peptide bond. How are they connected? They are connected with the peptide bond. Clear? They are connected with the peptide bond. So when there is a straight chain of amino acid, when there is this linear structure, this basic structure, here you will see N terminal. What is the meaning of N terminal? Your amino group is free here. Your amino group is placed first. This is the first amino acid and this one is the last. So here what will you see? You will see the carboxylic group here. So you will call it as C terminal, right? You will call it as C terminal. So this linear chain, this linear structure is basically the primary structure of proteins, right? What is it, bache? It is the primary structure of proteins. Is that clear? What is it? It is the primary structure of protein. So here you can see Frederick Sanger gave the primary structure of insulin for the first time. Okay, of insulin for the first time. So proteins, they may have one or more than one polypeptide chain. Now sometimes the students get confused here that what is the meaning of this. See, polypeptide means many peptide bonds are there. Like your amino acids are connected, they are forming these peptide chains. So it's not mandatory that one peptide chain is forming one protein. No. Sometimes many these peptide chains, many these polypeptide chains, they are joined together and they are forming the protein. Right? They are forming the protein. So amino acid of each polypeptide in a protein are joined to each other in a specific sequence, and this sequence is termed as primary structure of that protein. Okay, this sequence is termed as primary structure of that protein. Wow, Gopika, thank you so much. Clear, bache? So this is the primary structure, the straight chain, the linear chain, right? Right? Okay. So can I say that this primary structure, it describes, see, it is a primary structure. It's a linear chain. So here you can see first amino acid, which one is second, which one is third. So basically you can check the position of that amino acids, isn't it? You can check the position of that amino acid. Okay, bache? Right? Right? So you guys can check what? You guys can check the position of the amino acid. Okay, so here you guys can check the position, the structure, the type of that protein. Priyanka, last and second last, OH present on same side. But that is for the carbohydrate, na Priyanka? D and L configuration of amino acid. Soumyadeep was asking about it. Hai na Soumyadeep? D and L configuration of the protein is for amino acid. Hai na? Okay, so this is what you guys can write, that when it comes to the primary structure, the positional information is provided. So it describes type, number. It describes type, number, sequence, even the structure of that amino acid. Okay, so this is what you people can see when you talk about the, primary structure of protein. Done, bache? Done, bache? Okay. Any doubt? Yes, so it is a basic structure. It is the basic structure. So amino group terminal here, carbon terminal here. Not the structure. Oh, this is the order basically. Yes, order of amino acid. So this is the information that you are going to get from your primary protein. Clear, bache? Sure? Sure. So next is your secondary. Fine. Next is what? Next is your secondary structure. See, now when you are talking about these proteins, na, primary, secondary, tertiary, like this, so what you need to focus, like it is just like this thread. This thread, it's a straight chain, linear chain, primary structure. It will coil a bit, right? Again, you know, my mic. See, sorry, let's say it is coiling. It is showing some more bonding. It is not just having the one peptide bond, it is just coiling. Secondary structure, it is coiling more. Tertiary structure, it is coiling more. Quaternary structure, right? So that is how you are going to see this. Clear, bache? Clear, bache? So I cannot hold it much, otherwise, you know, na, mic kharab ho jayega. Okay? So next is what? Next is the secondary structure. Next is what? Next is the secondary structure. Clear, bache? So in secondary structure, we will cover alpha helical structure, or we will talk about the beta pleated structure, right? Beta pleated structure. That is what we are going to discuss here. So obviously here you will see the folding of that primary structure. What are you going to see in the secondary structure, bache? You are going to see the folding. right? You are going to see the folding. So here the folding and coiling of amino acid will be there. Folding and coiling of amino acid will be there. Are you getting it? What will you see? Folding and coiling of amino acid will be there when it is the secondary form of structure. Okay, bache? Okay, bache? So as I said, alpha helical and beta plated. Alpha helical and beta plated. Here I am showing the intramolecular hydrogen bonding. Wait, we will discuss. So this is the alpha helical structure. Always remember it is alpha. This helical. I hope you have seen that, you know, helical staircase. Helical staircase. You will see here this right-handed helical structure, right? This right-handed helical structure. So obviously, bache, now you are coiling or you are folding your linear chain. What are you doing? You are coiling, you are folding your linear chain. Okay, you are coiling, you are folding your linear chain when you are talking about the secondary structure. So obviously there will be some more bondings as well. Just in primary structure, there can be many polypeptide chains. It is not mandatory only one polypeptide chain will be there, but it's a linear structure where you can see the type, the number, the order of the amino acid, right? But you can see such, you know, the positions of that amino acids there. But now in the secondary, we are saying it is going to be, it will coil. In the secondary, what are we saying? It will coil, right? It will coil. So alpha helical structure, right-handed helical it is. Okay, right-handed helical it is. So obviously that protein chain is coiling. Now, what is going to happen here? Let's say here you have the first amino acid. This is just to explain you as of now. Okay. Okay, so let's say here you have your second amino acid. Here you have your third amino acid. Here you have your fourth amino acid. Here you have your fifth amino acid, something like that. So here the first amino acid and your fourth amino acid, it will show intra. Why intra? Because it will be there in the within the molecule. So it is intramolecular hydrogen bonding, right? It is the intramolecular hydrogen bonding. What will you see here? Intramolecular hydrogen bonding. Like the first, basically the bonding is in between the first amino acid or the fourth amino acid. Together they are showing the hydrogen bond formation. So we are calling it as intramolecular hydrogen bonding. Intramolecular hydrogen bonding. Let me show you this. See. Amino group here, and this is the fourth amino acid, it's carboxylic group. That is how you will see this bonding in between this O and H. This is the hydrogen bonding. Okay, this is the hydrogen bonding. So alpha helix is one of the most common ways in which a polypeptide chain can form all possible hydrogen bonds by twisting into a right-handed helix or right-handed screw. This point is very important, right? MCQ can come from this part. Okay, so this alpha helix is what? It is right-handed. This alpha helix is what? It is right-handed. Clear, bache? Clear, bache? So it will show the intramolecular hydrogen bonding. Okay, intramolecular hydrogen bonding. So NH group of your each amino acid, it will form hydrogen bond with the carboxylic group of an adjacent turn of the helix. Right, adjacent turn of the helix. So obviously turn. So I'm just telling you about the position. So here we will see one turn, right? Here we are going to see one turn. So first and the fourth, the language is different, but ultimately the meaning is same. Okay, so stabilization of this alpha helical structure is majorly because of the hydrogen bonding. So it is also important. So it is the intramolecular hydrogen bond. Clear, bache? What is it? It is the intramolecular hydrogen bonding. So intra means within. This is what you need to remember. Any doubt? Any doubt? So yes, examples mein you can see the keratin, which is the dead protein in our skin, in our nails. Your myosin in muscles is also the example of alpha helical structure. Fibroin in silk. So alpha helix, this information is not required, leave it. Take it. So next is the beta pleated structure. Beta pleated. Here you will see different polypeptide chains. You will see how that polypeptide chains are, you know, they will join together. They will join together. Like let's say, here I have this one polypeptide chain. This is the next polypeptide chain, right? Here I have this one polypeptide chain. This is the another polypeptide chain. Okay, so they are joined together. So how will they join? How will they join? They will join with this intermolecular hydrogen bonding. This intermolecular hydrogen bonding. Now it is not intra, it is inter in between these two polypeptide chains. And sometimes it's not always like two, you know, different polypeptide chains will be there. No, sometimes that chains are folded in the form of pleats. You know, pleats in the skirts. In the skirts we used to see pleats in the saris, right? Have you seen a girl in a sari? You can imagine any color, red sari, black sari, blue sari, whatever it is. So I think you all have seen that pleats here, right? In the front portion, some pleated structure is there. Some pleats are there. So you know about it. Angie, do you know about it? So that is the pleated structure. So here you can see these pleats. Okay, so they will, intermolecular hydrogen bonding will be seen here. Okay, bache. So in beta pleated structure, all peptide chains are stretched out to maximum extension. Then they lay side by side, which are held together by intermolecular hydrogen bonding. Kindly don't make mistake here, right? So we are going to stretch it in this way that we will get the maximum extension, and that is how we are going to form it. Okay, that is how we are going to form it. Clear, bache? Dhoti, whatever you want to imagine you can, but I hope you are getting the meaning here. So when the size of the groups, like your alkyl group, is moderate, polypeptide chain contract a little and you will get beta pleated structure to protein molecule, as in silk protein fibroin. So fibroin is also the example here. Okay, silk protein fibroin is the example of this beta pleated structure. Clear? So they can be parallel, they can be anti-parallel. If all the polypeptide chains in same direction, the parallel, otherwise anti-parallel, right? Otherwise they are anti-parallel, right? So your keratin in here, it is the example of parallel conformation of beta pleated structure. Anti-parallel mein your silk protein fibroin is there, right? Anti-parallel mein your silk protein fibroin is there. Clear, bache? So keratin, myosin, they are your alpha helical structure. Even your tropomyosin, you know, tropomyosin, troponin, they are also your alpha helical structure. So it's right-handed. Done, bache? It's right-handed. Done. So any doubt? So silk protein fibroin is the example here in beta plated structure. Okay. Okay. Fine. Fine, fine, fine. Fine. So this is about the structure, right? So next is your tertiary structure also, but before that, see, in the first class also I asked you this question. Let's say they have given you certain chemicals or certain molecules, or you know, in the NCERT, in the NEET, let's say there is a question, they have given you so many names and they are asking you, out of the following, which one is biomacromolecule, which one is biomicromolecule. So you should know the examples. Okay, you should know the examples. I say, I hope you know about the collagen. Do you know collagen? Collagen is the most abundant protein of animal world. It is the most abundant protein of animal world. So if it is a protein, then obviously it will be the part of acid insoluble fraction, isn't it? It will be the part of acid insoluble fraction, isn't it? Collagen. So it is triple helical. What is it? It is triple helical. It will have 3 chain. So it is rich in your, you know, proline, hydroxyproline, like this. Even glycine is there in collagen. And let me tell you, bache, this collagen is also the part of extracellular matrix in the case of animals, of course, right? It is the part of major component of extracellular matrix. Jaise we discuss it now, jaise one cell, second cell, third cell in our body, we are the multicellular organism. So obviously these cells are connected together. So here this extracellular matrix is also rich with this collagen. Okay, it is also rich with this collagen. Again, important it is, right? Important it is. Done, bache? Yes. Is it clear? Is it clear? Sure. So next is your tertiary structure. So obviously now the folding will increase, right? The folding will increase. Primary, secondary, tertiary, quaternary. Okay, quaternary. Done, bache? So tertiary structure, it is going to represent the folding of polypeptide chains or folding of secondary structure of protein. Fold your secondary structure more, you will get tertiary structure, right? You will get tertiary structure. So here you people can see the diagram of tertiary structure. You are going to see the hydrogen bondings there. You are going to see the hydrophobic interactions there. You will even see the disulfide bridge, right? Do you know that disulfide sulfur? I am repeating this again. Cysteine and methionine is the sulfur containing amino acid. Okay, it is the cysteine, it is the methionine. These two are the sulfur containing amino acids. Sulfur containing amino acids. Are you getting my point? Yes. Are you getting my point? So tertiary and secondary both are stabilized by the forces of hydrogen bond, but here you will also see disulfide linkage, van der Waals interactions, electrostatic forces of attraction as well. Fine, bache? Fine, bache? So tertiary structure, see, such enfoldings are going to be there. Such enfoldings are going to be there. So when it gets folded, yes, when secondary structure it gets folded, it will form tertiary. So the folding is in such a way, you will see the globular proteins here, right? You are going to see the fibrous and the globular proteins there. Clear, bache? Fibrous and the globular proteins there. Clear, bache? So here your side chains, right? It is basically the result of your interaction of the side chains, right? Because of the interaction of the side chains. Fine. Fine. So it is a 3D structure. Okay, it is the 3D structure. So obviously, see, now the side chains will interact. They will show the bonding. So when there is the interaction of side chains, that gives it a proper shape and function. Obviously, the interaction is in this way. You will get a structure, you will get a proper shape, and accordingly the function will be. So the tertiary structure is also considered as 3D structure of protein, the 3-dimensional structure of protein. And I hope you have read the NCERT. It is clearly mentioned, bache, that it is the biologically active protein. Okay, it is biologically active. Now, what is the meaning of being biologically active? Tell me. Biologically active. Your enzymes, right? When you talk about the enzymes, right? Even we will discuss it in detail as well. So when you people talk about the enzymes, you know that mostly enzymes are protein, yes or no? Mostly enzymes are proteins, isn't it, kids? Mostly enzymes are proteins, isn't it? Isn't it? And you know that these enzymes are the biocatalysts. They are going to catalyze the reactions in our body. So obviously, these enzymes, they are present in the 3D form. Their structure is 3-dimensional, right? So that is why we are saying tertiary proteins, right? They are having this biologically active form. They are functional in this way. This is the best example, okay, to understand this. This is the best example. So the tertiary structure. So ester forces are the ester bonds are the bonds which are not playing any role here in the stabilization of this tertiary structure. So you have seen the bondings here. So it is going to stabilize the tertiary structure. Clear, bache? Clear, bache? So as I said, you will be having the two major form, fibrous and globular. And if I need to talk about the major example here, so one is enzymes and another is myoglobin as well. You know that in the muscles, that oxygen carrying pigment myoglobin is present. So myoglobin is also the example of tertiary protein. Your enzymes are also the example of tertiary proteins, right? Your enzymes are also the example of tertiary proteins. Clear, bache? Clear? Sure, sure. So see this coiling, this further coiling, right? Just have a look, bache. Just see this further coiling, the bonding. So see, it is alpha helix. Ultimately, I told you, the secondary structure will get coiled more. So see, this is the alpha helix having intermolecular hydrogen bonding. These are the beta pleats having intermolecular hydrogen bonding. Now we are coiling the structure. Random coils I'm trying to show you. So see, the hydrophobic interactions are also there, right? Sometimes see the salt bridge and all. Your HSP sir is going to explain this part, and Wazim sir. Hydrophilic interactions you people are going to see. Covalent bonds you guys are going to see here, right? So that is how they are interacting. So these bondings are responsible for the stabilization of the structure. Clear? These bondings are responsible for the stabilization of structure, and the nature majorly it depends upon the amino acids, that what type of amino acid is there. Done? Any doubt? Okay, any doubt? So myoglobin, the best example to remember. Bache, we are about to finish the proteins. So after Deepa bache, you can check our channel, you will get an idea of research class. Okay. Right now focus here on biology. See, after completing the proteins, I'll give you a break for 15 minutes. Then we'll talk about the lipids, the nucleic acid, and then break, and then enzymes. So it depends upon you. The way you want, we'll carry it forward. Okay. We are going to carry it forward. Done? Nahi, Nikhil. Okay, so quaternary structure. Next is what? Next is the quaternary structure. Okay. Quaternary structure, quat, 4. The best example that you need to remember is hemoglobin. So here you will see like many polypeptide chains, they will become spherical, and many spherical structures, they will come closer and they will form a structure. Like here you guys can see 1, 2, 3, 4 polypeptide chains are there, and see they are coiled, they are forming these tertiary spherical structure, and they are coming closer. So some of the proteins, they have more than one polypeptide chain, right? So if they have more than one polypeptide chain, to each chain you people will call subunit. Right? So here we have more than one polypeptide chain, and you can call these chains as the subunits. So when these subunits, they have spatial arrangement with respect to each other, then they are going to form the quaternary structure. So the best example that we study is of hemoglobin. Okay. So what is hemoglobin? Hemoglobin is our respiratory pigment, isn't it? Hemoglobin is our respiratory pigment. So it is present in RBCs. Okay, it is present in RBCs, in your red blood cells, in your red blood corpuscles. So when you talk about these hemoglobin, they have 4 polypeptide chains. How many chains are there, bache? 4 polypeptide chains are there. 4 polypeptide chains are there. And each chain is what? Each chain is your subunit. Fine. Each chain is what? Each chain is your subunit. Done. So we have 2 alpha, 2 beta chains here. 2 alpha, 2 beta chains here. Clear? 2 alpha, 2 beta chains are there. So in the hemoglobin, if you remember, the globin is majorly the protein part. Okay, it is the globin which is the protein part. Heme is the pigment having iron. Basically iron as central metal, iron as central metal. It is the iron as central metal. Remember? Heme. So here also you will get one heme. So obviously this heme will bound to the oxygen, and oxygen. So when one hemoglobin is fully saturated, it will carry 4 oxygen molecules. Right, bache? It will carry 4 oxygen molecules. Done, bache? Sure. Sure. So these are the quaternary proteins. So these are the subunits here. Fine. These are the subunits here. Each chain is what? Each chain is the subunit here. And along with subunit, you can also use the word protomer. That's extra. You want to note it down, you can. You want to leave it, you can. It's your wish. Fine. It's your wish. Done, bache? So now just see. So that is how these foldings are present. Straight chain, then coiling alpha helical. It can be beta pleated also. Further coiling will form the tertiary structure, and then the aggregation of 2 or more polypeptide can form the quaternary structure. So any doubt? Doubt? Neonatal mein it's not. there is a in the neonatal. It's a good question. Adult hemoglobin is different from the fetal hemoglobin. Okay, fetal hemoglobin is adapted to. It is more efficient for the oxygen. Okay, even if oxygen is less, fetal hemoglobin will quickly bind to it. Okay, so now that's your homework. You will tell the differences in the fetal hemoglobin and in the adult hemoglobin later after the class in the comment section. Clear? In the comment section, this is what you guys are going to put. Now, remember one thing, bache, when it comes to the enzymes, you know, enzymes are highly specific. So that specificity, that just say, you know, the primary structure. You guys can see your asparagine, your glycine, phenylketonuria, glutamine, glutamic acid, glutamine, alanine, arginine. So see, there is a sequence of amino acids. There is a sequence of amino acid, proper sequence, proper position is there. So this specificity is due to the primary structure. Like how in the primary structure, the various amino acids are placed. This specificity is due to the primary structure. But right, this specificity is due to the primary structure, but biological activity is because of the tertiary structure. Right, so the biological activity is because of the tertiary structure. Is that clear? Is that clear? So it is because of the tertiary structure. Done, bache? So this is about the proteins, and then we have to discuss this on the basis of shape. Just allow me a minute. Done, bache? Just a minute. Good evening, good evening. So if you are a new student, do subscribe our channel. Fine. So the next is on the basis of shape. So on the basis of shape, obviously it is fibrous or globular. So globular like this, folded. Fibrous is going to be straight like this. So when it is the fibrous, the example is keratin, myosin. And in the case of globular, you will see the example of insulin. You will see the example of albumin, enzymes. Even your hemoglobin is also the example of your, yes, bache, it is also the example of your, yes. What type of protein? Globular protein. Clear, bache? So when it is fibrous, it is going to be thread-like, and when it is globular, obviously it is going to be rounded. It is going to be folded. Okay, and here it is thread-like. Okay, or you can say that rod-like structure. Fine, bache? You can say that rod-like structure. But always remember, right? So your enzymes. This is important. This is important. Your enzymes. Right, enzymatic activity is, you know, that tertiary structure is basically that enzymatic is biologically active. So your enzymes, they are globular. Okay, your enzymes, they are globular. So we can say that globular proteins, they are enzymatic. Okay, and when it comes to the fibrous protein, they are non-enzymatic. Fine, they are non-enzymatic. Is that clear? Yes, is that clear? Right, so globular proteins are comparatively unstable, right? But here you will see more stability. Done. You will see the stability. So examples are important. Fine, bache, examples are important. So keratin, myosin, your fibrous. So obviously the myosin protein, You know, na, it is used there in the muscles. So the next is on the basis of hydrolysis of protein. We can also do the hydrolysis of protein, na? Hydrolysis, breakdown in the presence of water, right? Hydrolysis. So on the basis of that, simple proteins are there, conjugated proteins are there, and the derived proteins are there. So majorly you need to know about the primary, secondary, tertiary, and quaternary, but at least you should know these terms. Okay, you should know this term. So when it is simple, see, hydrolysis of protein. What's the point? Hydrolysis of protein. So obviously breakdown, on the basis of breakdown, yeah, how are we forming that structure? It depends upon that, right? How are you forming that structure? It depends upon that, right? So albumin, globulin, simple proteins, conjugated mein you know that when to the protein carbohydrate part is attached, like glycoproteins are there. If nucleic acid is attached to the protein, nuclein is there, right? Chromoprotein obviously related to the color. So hemoglobin and chlorophyll, it is also the example of proteins, but they are conjugated protein. And further, right, from this, when these conjugated proteins, right, when they are hydrolyzed partially, you will get the derived proteins like your peptones. Even proteoses are also there. Even proteoses are also there. So it is little, you know, extra, but it is ultimately conveying everything. Fine. So you use these terms and you see these terms in NCERT. Clear, bache? You see these terms in NCERT. Done? Sure? Sure. So next is the denaturation of protein. Bache, denaturation of protein is a very simple topic. Denaturation of protein. It's a very simple topic. You know the primary structure, you know the secondary structure, you know the tertiary structure. Let's say when it is the denaturation, ultimately the structure of a particular protein will be changed because of the change in the temperature. Let's say if you are increasing the temperature, if it is a tertiary structure, increase in temperature is going to break the bonds. So if it will break the bond, obviously the structure will change. Accordingly, the activity will change. First point. This is the denaturation. Like I'll give you the example of the enzymes. We used to say that enzymes are highly specific if you increase the temperature, right? So in the high temperature, what is going to happen? In the high temperature, there will be the denaturation of the enzymes. Okay, why? Because in tertiary structure, the bond will get break, right? And the structure will change, of course. Okay, the structure will change and now it will not be biologically active. This is the example of denaturation. And obviously when temperature is low, denaturation will not be there, enzyme is not active. We will discuss it later. Okay, but this is the denaturation. Now, in the case of egg, you know na, egg white is basically having albumin protein. Albumin protein. So when we form that omelette, so in that omelette, the changes that we see, that coagulation of protein is also showing the denaturation, right? When you make that omelette, na, you break that egg, so that white part, it will get converted, so that coagulated white part is also showing the denaturation of protein. Clear, bache? It is also showing the denaturation of protein. Clear? Yes? Is it clear? Done? So coagulation of egg white on boiling is a very common example of this denaturation. And curdling of milk. Curdling of milk. You know, na, milk is having a protein, casein. Milk is having a protein which is casein. So what is going to happen? See, ultimately in the milk, we have the sugar as well. So lactose is the sugar. It's a disaccharide which is present in milk. Students, they make the mistake here, right? So when you talk about the milk, milk is having the lactose. Lactose is what? Lactose is the sugar. Lactose is the disaccharide there. When you use the word casein, casein is the protein which is present in the milk. Okay, casein is the protein which is present in the milk. So now the bacteria, I hope you know the name of that bacteria, lactobacillus, right? That is lactobacillus. Okay, so this bacteria will act on this milk protein will act on this lactose. It will convert this sugar into lactic acid, right? Because of bacterial fermentation, this lactose will be converted to lactic acid. Now this lactic acid is responsible for the coagulation of the milk protein casein. Are you getting it? It is responsible for the coagulation of the milk protein casein. And that coagulated protein, obviously this is basically the curdling of milk. We will get the milk. the curd. Fine, we will get the curd. Done, bache? Done, bache? So whenever there is any, right, change, whenever there is any physical change in the protein structure, right, it can be due to high temperature, and even it can be because of change in pH. It's not always you have to mention high temperature and pH. You can simply write change in temperature, change in pH. So change in temperature and pH can cause the change in physical structure of the protein. That is basically the denaturation. So tertiary structure can become linear. So obviously it will not behave properly. It will not do its work. Clear, bache? It will not do its work. Done? Done. So globular protein can become unfolded, helix can get uncoiled, and protein can lose its biological activity. This is basically the denaturation. So during denaturation, secondary and tertiary structure, they will be destroyed, they will become primary. Okay, they will be destroyed, they will become primary. Clear, bache? So this is all about the denaturation of the proteins. This is all about the proteins. So let's revise it from NCERT, and then what? Then obviously let's take a break, and we'll continue then. Arey, go slow. Proteins. See, so let's start from the amino acids first. Okay, so now it's time for the NCERT reading. Okay, so amino acids are organic compounds having one amino group and an acidic group. True. And these are as substituted on the same carbon, that is the alpha carbon. Any doubt here? Are you able to understand it now or not? Can you understand it now? I think you can. So they are what? They are the alpha amino acids. Right? They are what? They are the alpha amino acids. So they are substituted methanes. You know na, methane CH4. And now when you're talking about the general structure of protein, this is what you are going to get. So obviously it's a substituted methane. And if this carbon is attached to 4 different groups, right? So it's the alpha carbon, it is the asymmetrical carbon, right, bache? Right? So there are 4 substituents group occupying the 4 valency positions. So these are hydrogen, carboxylic group, amino group, and a variable group. Right? So based on the number of R group, we have different type of amino acids. So those which occur in proteins, they are mainly 20 types. So you know about these 20 amino acids now. Clear, bache? And see, this is the zwitterion which is given in your NCERT. Fine? Fine. So R in these proteinaceous amino acids, it can be hydrogen. So if only hydrogen is there, it's glycine. If methyl group is there, it's alanine. If hydroxyl methyl, that is your CH2OH is there, it is serine. So yes, you have to revise it. Right? You have to revise it. Clear, bache? So the chemical and physical properties of amino acids are essentially of the amino group, carboxylic group, and of this functional group. So on the basis of, number of amino acid and carboxylic group, we have acidic, right? So in acidic amino group, you know that you have 2 COH group and 1 NH2 group. Clear, bache? So when it is basic, you know that you have 2 NH2 group and 1 COH group, right? So net charge is going to be positive. Net charge is going to be positive. So lysine is the example. And when it is neutral, all 15 amino acid, and as per NCERT, it is the valine. Okay, so aromatic are tyrosine, phenyl, and tryptophan. I told you already, bache, right? And the ionizable nature, they are having the ionizable nature. Hence, in solutions of different pH, the structure of amino acid changes. Fine. So I hope you know about these zwitterions now. Clear, bache? Clear? So the next part to it is proteins. So here you guys can see proteins are polypeptide. Yes, ma'am. So they are linear chain of amino acid linked by peptide bond. So what is your peptide bond? C double bond O and H. That is your peptide bond. Okay, that is your peptide bond. So bache, once there was a question from this part of NCERT as well. So GLUT4, right? Once they asked the question from this part, they asked the GLUT4, and many students were like, ki, bhai, it is out of NCERT. Actually, it is not. So tables here, especially in the biomolecule chapter, is very important. So collagen, intercellular ground substance, trypsin, enzyme it is, insulin, hormone it is, antibodies which fight with the infectious agent, receptors for smell, taste, hormone, and GLUT4 enables glucose transport into the cells. Clear? So the examples are important, right? So GLUT4 is nothing, it is the transporter. Sometimes the glucose cannot cross the cell directly. So it is what? It is the transporter there. Fine, bache. It is what? It is the transporter there. Clear? So each protein is a polymer of amino acid. Fine. Right. So a homopolymer is having only one type of monomer, but in protein different amino acids are there. That is why it is heteropolymer. Clear, bache? That is why it is heteropolymer. Right? So you know that dietary proteins are basically the essential amino acids. So amino acid can be essential or non-essential. I told you already. Fine, I told you already. Done, bache? So collagen is the most abundant protein in animal world. This is very important. And another is rubisco. You remember na, rubisco? It is ribulose. It is the most abundant protein of biosphere. It's ribulose, ribulose, bisphosphate, carboxylase, oxygenase. Okay, carboxylase, oxygenase. Fine. Yes, bache, done. So ribisco is ribulose, bisphosphate, carboxylase, oxygenase. So it is the most abundant protein in the whole of the biosphere. So now you know that it's ribulose, right? Ribulose. Ul is there. Ul is there. Matlab it is a ketose. Okay, it is a pentose ketose. Fine, it is a pentose ketose. Done, bache? So that's about the proteins. Anything else that we need to discuss from NCERT? No, not yet. Okay, so let's discuss the protein, but after the break. So there will be the break time. Okay, so now you guys are going to get the break. Fine, and then we will discuss it. We will continue it. So we will meet at 8:15. Okay, right? Visual proteins में rhodopsin, idopsin, pigments, they are basically. Okay, so let's meet after 15 minutes. Priya, ask Avenger batch students. Anatomy is too easy. I'm sorry, I am late. Why? Because I didn't get my tea on time. That's why. So how are you guys? Welcome back. Welcome back, welcome back, welcome back. So now we will talk about the lipids and the nucleic acids. Okay? Okay? So tell me, how are you doing, people? How are you all doing? Good. So can we start? Priya, that's why you need my lectures. It's okay. Guys, first of all, let me tell you one thing. We are soon going to close the entries for the NEET Energized batch, and it is all obviously it is a very good decision. Why? Because you know, if you will join late, you'll cover your syllabus late. Again, you will keep on asking, ma'am, what to do with the backlogs, how to complete the backlogs this, backlog that, right? Again, you will be under stress and you will not prepare well. So that's a very good decision by company that we are going to close the entries for that particular batch. So still, if you want to join, most welcome, because from 1st of August, I'm going to start PP, that is your plant physiology, human physiology, genetics, reproduction, biotech chapter complete. Take it. So the most important chapter of class 11th we are going to start, and in the month of November you will get the 10 to 15 days for the revision, right? I will not start any new topic, and in that time period you guys can cover your backlogs. Okay, so how to join the batch, let me show you guys. So you can see here in the description box of the video, you have to click on this NEET Energized English batch link. Okay, then add your number, add your details, and you are going to get this batch at a price of 14999. Right? Okay, and just one student is asking for the anatomy. So in the same way, bache, yes, and the same way, see, today I have posted one video that is life cycle of plant kingdom. So obviously your 4 marks are confirmed here, and here we have covered all the important things related to the your life cycles. Okay, so what I want to say is, please support this short series. Only then I will be able to post more videos, right? And I want to post the videos till your NEET examination, so that you will get a, you will get a proper playlist where all the important questions, all the important topics are covered in less than 30 minutes, right? I will try my best to complete it in 10 minutes, but if some topics are lengthy and you need to, cover everything of that topic, so obviously it will take 20 minutes. So in less than 30 minutes, we need to cover the concepts. So imagine if I'll start now, till NEET 2024, we can complete our entire biology in this way, basically all the important questions. Okay, so with the help of the short series itself, easily you guys can answer 50 questions very easily. Directly you will be able to score good marks in 40-50 questions, and you just need to give the 10 to 15 minutes of your day. But I need support here, right? Please watch them, please put the comments, please share it with your friends, right? Please share it with your friends. Okay, so life cycle of plant kingdom, do check it out ASAP. Fine, chalo. So now let's go to the lipids part. So now you know that lipids, they are not the bio-macromolecules, isn't it? Lipids are not the bio-macromolecules. Clear? Just a minute, guys. Okay. So now when you talk about the lipids, no doubt they are what? They are not polymeric. Lipids are not polymeric, and these lipids, right, but still they are present in the acid insoluble pool. They are bio-micromolecules, but they are still present in the acid insoluble pool. Clear, bache? In the acid insoluble pool they are present. Now, why are they in the acid insoluble pool? Obviously, it's very simple, bache, because you know that lipids are water insoluble, right? Lipids are making the plasma membrane of the cell. Okay, the adipose tissue is covering the soft organs of our body. It is providing the protection. It is acting like a shock absorbent. So lipids are important in our body. So whenever we take any living tissue, we break it, right? We add the trichloroacetic acid there and we try to separate it. So obviously, because these lipids are insoluble in water, they are going to form the vesicles and they will not pass in that acid soluble pool. They will retain there in the acid insoluble pool. Basically, they will be there in the retentate. Clear, bache? They'll be there in the retentate. So this is the story of the lipids. So if I talk about the most common type of lipid, so we are going to talk about the triglycerides. What are we going to discuss, bache? We are going to talk about the triglycerides, right? Triglycerides, most common type of lipids. So now here you guys can see it is from NCERT. Your fatty acids, your glycerol, your triglyceride, it is given. So now this is what we need to understand, this is what we need to discuss. But first of all, you people tell me, what is the difference in between you can say that, yes, what is the difference in between a fat and a lipid? What is the difference in between the fats and the lipids? Do you know that? Any difference in the fats, in the lipids? Quick. Any difference in the fats and the lipids? Typical fat molecule, what? Tell me, what is the difference there? Head, what else? See, ultimately lipids are also the organic compounds. Fine. Ultimately, these lipids, they are also the organic compounds, and these lipids are having carbon, they have hydrogen, they have oxygen. Now, when you compare, right, when you compare it with the carbohydrates, in the carbohydrates, when we were talking about the carbon, hydrogen and oxygen ratio, in the carbohydrates, we were discussing that, yes, can you tell me what was there in the carbohydrates? We used to say na carbon is there, hydrogen is there, oxygen is there. If you remember, yes, we used to say na one part of carbon, two part of hydrogen, one part of oxygen. But here in this case, right, when you are talking about what? When you are talking about the organic compounds, about the lipids, so in their case, the ratio of H and O, right, the ratio of hydrogen and oxygen is not 2:1. Like in the carbohydrate, it was 2:1, but here it is not going to be 2:1. This is the important point. And actually, let me tell you, bache, from the fats, we used to get more energy than the carbohydrates. But still, we say the carbohydrates are the major respiratory substrate. Why? Because they will be easily oxidized. And for the oxidation of these fats, a lot of energy is required. So that is why this is the point here. Okay, this is the point here when you talk about what? When you talk about the lipids. So now you have to draw one flow chart. So here, in the case of lipids, we also discuss simple lipids. We discuss compound and conjugate lipids. And we also talk about the derived lipids. Fine. We also talk about the derived lipids. This is what we used to discuss. Now in these simple lipids, right, when you are talking about these simple lipids, they are simple esters of fatty acid with alcohol. I'll explain it as well. So they are simply the esters of fatty acids with alcohol. And here what are you going to see in the simple lipids, bache? You will see your true lipids, right? And you will see the waxes. Like you will see the neutral or the true fats. And here you are going to see the waxes as well. Now the question arises here, ma'am. See, I will explain this line that what is the meaning of esters of fatty acid with alcohol. No doubt I'm going to explain it. But as of now, just draw this, right? Just draw this flow chart. So when you are saying it is a neutral or, when you are saying it is a neutral or true fat, so it means it is the esters of fatty acid with glycerol. So the alcohol present here is glycerol, right? Alcohol present here is glycerol. And when you are talking about the waxes, right? So here what will you see? They are also the esters, no doubt. Esters of fatty acid with monohydric. Mono means single, hydric means OH, with monohydric alcohol, right? So you talk about the ear wax, you know na ear, it is having some yellowish kind of wax, cerumen, ear wax, right? Right, we used to put that earbud and we used to take it out, some yellowish, that is cerumen, ear wax. Right, so this is a simple lipid when you are talking about the esters of fatty acid with the alcohol. So majorly, right, in the neutral or true fats, that alcohol is glycerol, but it can be monohydric alcohol as well, right? Monohydric alcohol as well. Okay, so now when it is the conjugate lipid, what is it? It is a conjugate lipid. Again, they are also the esters of fatty acid with alcohol, but along with alcohol, right? So these are also the esters of fatty acid with alcohol, right? They are what? They are also the esters of fatty acid with alcohol, and extra functional groups are also there, right? Extra functional groups are also there. So like you have the best example of your, yes, you have the best example of your phospholipids. Remember? So phospholipid is the example of your compound lipid, of your conjugate lipid, where phosphate group is also there, right? Where phosphate group is also there. So it is not just the phospholipid. You have another examples also, right? Glycolipids are there, lipoproteins are also there. Clear, bache? Glycolipids are there, lipoproteins are also there. Now when it is derived, they will have the rings and the long hydrocarbon side chains, right? They will have the rings, hydrocarbon rings, and long hydrocarbon chains as well. Clear, bache? Long hydrocarbon chains as well. So that is what we need to study here, right? So lipids, they are the biomicromolecule. Their molecular weight is not less than, above 800 Dalton. It is less than 800 Dalton, but still it is the part of acid insoluble pool. That is your retentate. Clear, bache? That is your retentate. So we have simple lipids, right? The conjugate or the compound lipids are also there, right? The conjugate and the compound lipids are also there, and then comes the derived lipids. Clear, bache? Then comes the derived lipid. Done? Done, bache? So they are insoluble in water. Clear? They are insoluble in water. So ultimately, fatty acid will be esterified with the glycerol. Clear, bache? They will be esterified with the glycerol. Clear? Clear? So, okay. So you need to understand one thing here. See, glycerol. Glycerol is also considered as simple fat. Okay, glycerol. It's trihydroxypropane. What is it? It's trihydroxypropane, right? Trihydroxypropane. Carbon, carbon, carbon. Propane it is not carbon, carbon, carbon. Trihydroxy. So OH, OH, OH. So rest will be C, CH2, CH2, CH2. Clear? So this is the glycerol. That's all. Right? Its structure is as simple as that. This is your glycerol. Clear? This is your glycerol. It's a very simple structure. You all can see it here. Isn't it, kids? You all can see it here. It is a very simple structure. This is your glycerol. So tri means three hydroxy, three hydroxyl group, OH, propane it is. 3 carbon will be there. That is your glycerol. Now, when I use the word fatty acid, okay, when I use the word fatty acid, see, one thing is very simple here. Acid is the word. Okay, what is the word here? Acid is the word. So if it is fatty acid, obviously the functional group is going to be C double bond OOH. Isn't it? For sure there will be the functional group, and functional group will be C double bond OH. Isn't it, Priyanka? The functional group is going to be C double bond OH. Why? Because it is the fatty acid. Now, other than that, let's say you have, okay, C double bond OH, and here you have CH3. This is also your fatty acid. Okay, this is also your fatty acid. Can you name it? Can you name it? Okay, that's your homework. You have to name it. Fine. And here again, let's say it is again CH3. C double bond OH is confirmed. So add one more CH2 here. So that is how you can increase the CH2 number here in between, right? CH2 number here in between, and you will get the fatty acids. What will you get here? You will get the fatty acids. Are you getting it, bache? Yes, bache. Are you getting it? So glycerol, trihydroxypropane, and here you guys can see the structure of the fatty acid as well. So if there is any doubt, I'm asking it again, bache, please let me know. Okay, if there is any doubt, just let me know. So this is basically your fatty acid. So here when you will see the number of carbon in totality, okay, this is what we are going to discuss here in that fatty acid chain. So can I not say that it is the chain of carbon, hydrogen, oxygen? So basically it's a chain of carbon, hydrogen. Carbon, hydrogen is added there. The C double bond OH group is added there. So carbon, hydrogen chain it is now. And at the end you will see that this is the functional group here. Right, this is the functional group here, isn't it? So here in these fatty acids, when you will see the number of, right, the number of carbon, so you can keep it from 1 to 20, right? The number of carbon, I'm not talking about the, right, I'm not just talking about this carbon hydrogen chain. Overall, I'm saying the number of carbon is going to be 20. Just in NCERT, it is written like that in the carbon hydrogen chain, the number of carbon is going to be 1 to 19. Right? Here it is mentioned as 19. But overall, I'm talking like, let's say here 2 carbon and 1 carboxylic functional group. We can say it like this also, that here 2 carbons are present and 1 carboxylic acid group is attached to this fatty acid. Or we can simply say that number of carbon is 3. It's all same. Ultimately, the meaning is same here. Isn't it, Karthik? The meaning is same here. Right? So this is what we need to discuss. This is what we need to understand now. So when you are talking about the fatty acid, ultimately one thing is for sure that you are going to get the C double bond OH, the functional group, and to this that carbon hydrogen chain is attached. So keep it as alkyl group. So ultimately it is the alkyl group. So the number of CH2 groups will be changed accordingly. Okay? Okay? So now this is your glycerol. Now we are saying that fats, especially the true fats, are esters of fatty acids and glycerol. That's what we are saying. The true fats are the esters of fatty acids and glycerol. That is what we are saying. Now when it is a fatty acid, just write it like this, C double bond OH. Okay, and just keep one R1. So now we have to make the ester bond. What we need to make, bache? We need to make the ester bond. So from this part, right? From this part, what will go out? The water, the H2O. Now, if you will ask me that, ma'am, which oxygen will go out, right? This one of glycerol or this oxygen of fatty acid? I am not the right person to answer it, to be very honest. You can ask this question from HSP sir. He will explain it with the meaning. Okay, that madam, because of this reason, right, this OH will go out. And I have read it somewhere, I don't remember exactly right now, that which oxygen will go out actually when the water will go, when the water will form. There is a reason behind it, but right now I'm not able to recall. Okay, so water will go out. So obviously, let's say if water is, if water will go out here, 2H and 1 oxygen. So you are going to left with this part, na? C double bond O and acha, theek hai, H here, isn't it? This is what you are going to get. This is what you are going to get. Let me draw it once again. OH and here OH. So what I just said, what will come out? The water will come out. The water will come out. So obviously 2H and 1 oxygen will go. Okay. 2H, even I was 2H and 1 oxygen will go, right? 2H and 1 oxygen will go. So you are left with this. Isn't it? Isn't it? Yes or no? Isn't it? C double bond O, O. This is how it is attached. This is the ester bond now. Yes or no? And if you have to make phosphoester bond, that we will study in the nucleic acid, so it is going to be like this. This will be your phosphoester bond, and this is your like simple ester bond. I don't know whether we call it as carbon ester bond or not. I don't know. But yes, this is your phosphoester bond. So are you getting it? Are you getting it? Yes. So to this glycerol, which is trihydroxypropane. Stone bache, glycerol which is trihydroxypropane having 3 OH group. So here when you look at the bond formation, the ester bond formation, this is how the bond is going to form. Clear? So now one fatty acid is attached to the glycerol. So when one fatty acid is attached to the one glycerol, obviously the bond is ester bond here. The bond is ester bond here. So you will use the term monoglyceride. It is. Okay. You are going to use the term monoglyceride. You are going to use the word monoglyceride. Muskan Mahi, I love you. I love you too, Muskan. Now please focus here. So when two fatty acids, they will join to one glycerol. Ultimately your glycerol is one only. Take it. Take it this part. So these fatty acids can be same, they can be different, it doesn't matter, right? It can be same, it can be different, it doesn't matter at all. Fine, it doesn't matter at all. Now, it will be when two fatty acids are attached to one glycerol, it is going to be diglyceride. It is going to be diglyceride. And when three fatty acids, they join with one glycerol, it will be triglyceride, the most common type of your, right, the most common type of your, yes bache, fat. Okay, so it's a true fat where glycerol is there. Okay, it is the true fat where glycerol is there. Clear? Clear? Any doubt? So this structure is a triglyceride. This structure is a triglyceride. Done? Sure? Are you sure? So next part that we need to understand is bache, the next thing that we need to understand is saturated fatty acid and unsaturated fatty acid. Saturated and unsaturated. So when it is saturated, you can see the straight chain, right? Just look at this, bache. Just look at this. The carboxylic group is there, and then carbon hydrogen chain. So that is why I was telling you initially that one carboxylic group, and on another side you have this methyl group, and in between you just need to add the CH2, CH2, CH2 and CH2. Right, you just need to add the CH2, CH2, CH2 and CH2. That is how it will work. Fine, bache, that is how it will proceed. Fine. So this is unsaturated. So here you guys can see the double bond. Unsaturation means presence of more than one bond. Right, right. So basically single or double or triple bond is present basically when we use the word unsaturated, right? So here you can simply see that carbon-carbon double bond is there. So wherever there is the double bond, you will see this kind of bending. Okay, this kind of bending. We use the word kinks for that too, right? We also use the word kinks, the bends, right? The same thing. So carbon-carbon. So in between carbon and carbon, more than one bond is there when you are talking about the unsaturation. So same thing, the unsaturated fatty acids, we even discuss in the structure of the plasma membrane when we talk about the fluid mosaic hypothesis. So we used to say unsaturation is also there, cholesterol is present, this is present, that is present. Why? That unsaturation, these bends, they are also playing the role in maintaining the structure, no? Right, they are also playing the role in maintaining the structure, no? So that is how it is present. So this is the unsaturation here. Clear, bache? Clear, bache? This is the unsaturation here. So saturation, unsaturation, saturation mein single bonds will be there, right? And here you will see the carbon-carbon more than single bond is also present. Clear? Clear? So your bache saturated fatty acids, na, they are non-essential. What is the meaning of non-essential fatty acids? Saturated fatty acids, they are basically your non-essential fatty acids. Can you explain the meaning of this non-essential fatty acid? Yes. What is the meaning of the non-essential fatty acid? Tell me, what is the meaning of the non-essential fatty acids? Yes, tell me. Anyone? But it is non-essential fatty acid, means it is synthesized in our body, right? It is not mandatory that we have to take it in our diet. It should not be a dietary supplement. Okay, it should not be a dietary supplement. It is the part of our, okay, it is the part of our body. Basically, it is synthesized in the body. Done? Done, bache? Clear, bache? Yes, body manufactures them. And when you are talking about the unsaturated, here you will see the examples of your essential fatty acids also. So now let's talk about the difference in these two types of fatty acids. So saturated fatty acids and unsaturated fatty acids. Clear, bache? Saturated fatty acids and unsaturated fatty acids. Do I need to mention the definition as well? So now I hope you guys can understand this. So when it is saturated fatty acid, so of course carbon-carbon single bond is there. Clear, bache? So carbon-carbon double bond will also be there, which will result in the bends. So as I said, they are what? They are the non-essential fatty acids. So they are synthesized in body, right, bache? There is no need to supplement them in our diet as well. And now, if I talk about these fats, you know, metabolically they are slow. Do you know that? Metabolically slow. Basically, they are less reactive. They don't take part in the metabolism. You know, na, metabolism, the chemical reactions which are going on in our body. What is metabolism? The chemical reactions which are going on in our body. So as I said, metabolically, what are they? They are slow, right? They are less reactive. So if they are less reactive, if they are not taking part in the chemical reactions, so obviously there are chances that they will get accumulated in the body. Yes or no? There are chances that they will get accumulated in the body. So obviously it will result in that obesity. है ना? It will result in that obesity. So because of these saturated fatty acids, we gain the weight. The excess of weight we used to gain. Okay? We used to gain. Done, bache? Done. So these saturated fatty acids, they are S for saturated, S for solid. So they are mostly solid at room temperature. Like the example of butter, like the example of ghee. Okay, so they are the part of what? They are the part of saturated fatty acid. Is that clear? Is that clear? Yes or no? Okay, so in the animals, we will see the high amount of saturated fatty acids. Fine. Done, bache? High amount of saturated fatty acids. So examples are your stearic acid and the palmitic acid. Right, your stearic acid and the palmitic acid, they are the examples here. So when it is the stearic acid, the carbon number is going to be, bache, 16. Right? Carbon number is going to be 16. And when it is the palmitic acid, the carbon number is going to be 18. So I think now you guys can, right, now you guys can draw the structure as well. One second. Yes, palmitic acid is 16, stearic acid is 18. Yes, it is the palmitic acid, 16 carbon, the stearic acid is having the 18 carbon. Done, bache? Done, bache? So these are the number of carbons, Now you can draw the structure and you can see it here also. Done? So see, fatty acid, palmitic acid, 3 plus 14, 15, 16, 17 and 18. So palmitic acid mein carbon number is 18. Okay, it is 18. I think I made one mistake, isn't it? Sorry, it is not 18, it's 16 again. So 14, 15 and the 16, my bad. So carbon number is 16 here. So see, you can easily draw it. What do you have to do? COH, CH3 and accordingly put CH2 group in between. Done? Right, so 14 will be here. Done, bache? So same way mein, if you have to draw the stearic acid, see carbon number is 18. So COH, CH3. And when it comes to CH2, it will be 16 here. Done? So this will be your stearic acid. Fine, bache? So here you guys can see the glycerol, trihydroxypropane and your triglyceride as well. Fine, your triglyceride as well. So see, so I hope it's clear to you all. Done? Yes, any doubt here, bache? So now when you are talking about the unsaturated fatty acid, you're talking about what? You're talking about the unsaturated fatty acid. So as I said, what will you see? You will see the kinks, you will see the double bonds. So if there is any doubt, do let me know. Right? Is there any doubt? Do let me know. Okay. So double bonds are present with single bonds between the carbon-carbon atoms. Fine. So they are also the essential fatty acids. So what is the meaning of being an essential fatty acid? That they are not synthesized by our body, so they should be the part of our diet, right? They should be the part of our diet. Okay, so essential fatty acids are also from these unsaturated fatty acids. Clear, bache? Clear, bache? I'll give you one example. Bache, see, Keshav, Avengers batch, the entry for the Avengers batch will get closed. So if you want to join it, you guys can. But later on, if you think that we will again start with new batch, no, there can be a crash course, right? There can be a crash course, but that I cannot surely tell you right now. ठीक है? ठीक है, ठीक है, ठीक है. Okay, so they are essential because they cannot be synthesized in animal body, so it should be there in our diet. Done, bache? So the lipids which are having more essential fatty acids. Okay, so the lipids having more unsaturated fatty acids. For unsaturated fatty acids, I'm using the short form now, UFA. Unsaturated fatty acids, they are liquid at room temperature. Right, they are liquid at room temperature. The example is your oils. Fine, the example is your oils. Done, bache? Done, bache? The example is your oil. So you can take any example. You can take the example of your mustard oil, very commonly used in kitchen, isn't it? Mustard oil. Now, obviously, metabolically, they are going to be very active. So they are healthy as well. So metabolically active. So obviously, they are more reactive. Metabolically active. So it means they are more reactive. So they are going to take part in the chemical reaction. So they will not get accumulated here in our body. Okay. Okay, they will not get accumulated here in our body. So no obesity, no cholesterol will be there. No obesity, no cholesterol. So that is why even if you will see the ads, I hope you guys have noticed, right? When they used to, yes, Fortune and all, they used to say now that this oil is having MUFA, PUFA. I hope you know about it. MUFA, PUFA, like when it is MUFA, MUFA, mono unsaturated fatty acid, means there is single unsaturation. Okay, mono unsaturated fatty acid. So if double bonds are more than one, okay, then it is going to be more than one. It is going to be PUFA. That is poly unsaturated fatty acids. Are you getting it? Poly unsaturated fatty acids. Done, bache? Yes. No obesity, no cholesterol, no disease will be there. So unsaturated fatty acids are basically healthy for us. Clear, bache? They are basically healthy for us. Done? They are basically healthy for us. Okay, so these are the essential. What are they? These are the essential fatty acids. So when you talk about these unsaturated fatty acids, so you have some examples. Okay, so in the case of unsaturated fatty acids, of course, we have the examples. So for the examples, there is a trick, bache, and that trick is OLA. That trick is OLA. What is the trick here? The trick is OLA. Right? It is OLA. O stands for oleic acid. L stands for lin, oleic acid. L stands for lin, olenic acid. These are the examples of your unsaturated fatty acid. A stands for arachidonic acid. Fine. A stands for arachidonic acid. Now, if I talk about number of unsaturations, like how many double bonds are there? Number of unsaturations. So 1, 2, 3, and 4. That's how this trick works. So it's Ola 1, 2, 3, 4. Right? It's Ola 1, 2, 3, 4. Clear, bache? What is it? It's Ola 1, 2, 3, and 4. Any doubt? Any doubt? So Ola 1, 2, 3, 4. Clear? Clear, bache? So if I talk about the carbon number, if I talk about the total carbon number, 18, 18, 18, 20. 18, 18, 18, 20. Done, bache? It's 18, 18, 18, 20. So what is the trick? Ola 1, 2, 3, 4. Ola 1, 2, 3, 4. So 18, 18, 18, 30, and 20. So oleic acid, linolenic acid, linolenic acid, arachidonic acid. Clear, bache? And out of it, your lallaha, this LLA, these are the examples of your essential fatty acids. Okay, these are the examples of your essential fatty acids. So linolic acid, your linolenic acid, and your arachidonic acid, they are the examples of what? They are the examples of your essential fatty acids. So I'm repeating this again. 2 bonds, 2 double bonds, 3 double bonds, and 4 double bonds are there. Carbon number is 18 here, carbon number is 18 here, carbon number is 20 here. So I think you guys can write down the formula now. Right? Isn't it? I think you people can write down the formula. Like if I have to write down the formula of your arachidonic acid, so carbon, hydrogen, oxygen. So ultimately see, it's C double bond OH. So oxygen is going to be 2. Okay. Okay. So carbon number is 20. You already know. So what about the hydrogens? What about the hydrogens? So hydrogen here will be 32. Can you tell me how? See, this is a very easy way to calculate the formula as well. You know the carbon number. You know that only 2 oxygen atoms are there here in the carboxylic acid. So hydrogen number, and here what do we know? That there are 4 unsaturations. So bache, wherever there is unsaturation, let me show you. Like see, if here you have 1 unsaturation, so you guys can see, right? If you compare here, 2 extra hydrogen, they'll go out, right? We don't have 2 extra hydrogens here because we have carbon-carbon double bond here. Same way, in arachidonic acid, 4 double bonds are there. So obviously you guys can say 2, 2, 2, 2, 8. 8 hydrogens will be removed then. Clear, bache? 8 hydrogens will be removed then. So this is how you have to remember the structure. Okay, bache? This is how you have to remember the structure. Clear? Clear, clear, clear. So this is the trick for you. So examples, Ola, oleic, linolic, linolenic, and arachidonic. And here essential mein La, linolic, linolenic, arachidonic. So these are the examples. So triglyceride is the most common type of fat, bache. So this is all about the simple fat, simple lipids, about the neutral lipids. So now we need to talk about the conjugate or the compound lipid, where the most common is your phospholipid. So if there is any doubt, please ask it. Yes, please ask it if there is any doubt. Yes, bache, if there is any doubt. Sure. So just come to this diagram. This is from NCERT now. So you guys can see the palmitic acid is given there, right? The glycerol is given there. The triglyceride is given here, right? And then comes this phospholipid lecithin. So first of all, just check this particular structure from this side, right? It is what? These are the phospholipid. So now you know that when you talk about the conjugate or the complex lipids, so you know that they do not just have glycerol, they do not just have fatty acids, they have another groups also, right? So now it is the phospholipid. So obviously it is having the phosphorus also, and some other organic compounds are also there, right? They are having the phosphorus also, right? And other phosphorylated organic compound is there. Like look at this part, right? So I can say that this is a phospholipid. phosphorylated organic compound. Phosphorylated means, like, look at this particular structure, right? So nitrogen is also present here. This is the most common type of phospholipid, that is your lecithin. Okay, it is the most common type of phospholipid, that is your lecithin. Fine, that is your lecithin. Okay, so here you people can see this part, this lecithin, N group is also there, CH3CH. So phosphate is attached to this part, this nitrogen containing part. Actually, this nitrogen containing part is known as colon. It is known as colon. So you can see the phosphate group is attached here. So that is why we are using the word phosphorylated organic compound. Okay, that is why we are using the word phosphorylated organic compound. So this is how it is present here, bache. Clear? This is how it is present here. Fine. So this is the phospholipid lecithin. Okay, let me draw this. So ultimately the story is same, right? Ultimately the story is same. First of all, take the glycerol. You have to take the glycerol, and then, so to this glycerol, of course, because ester bond is there, simply you can write R1, R2, and here it is phosphorylated. So I hope you all have seen the structure. Right? Yeah, there is no mistake, but yes, one CH2 we need to add here. That's the structure. Clear, bache? That's the structure. That's all. Okay, fine. So to this part, even your OH is attached. That's all. So it is the same structure. It is the same structure. You can see here. So one glycerol, two fatty acids, and this is the phosphorylated organic compound. This one nitrogen containing one is the colon here. That is why you are calling it as lecithin. Okay, that is why you are calling it as lecithin. Done, bache? Sure? Sure. So they are phospholipids or phosphoglycerides. So you will see the saturated and the unsaturated fatty acids both here. Fine? Yes? Tell me, bachcho, is that clear? Okay, so this is the lecithin having one glycerol, two fatty acid, and one, this one is the phosphoric acid here, right? One phosphoric acid and one nitrogen containing group, that is your colon. This is important for your NEET, bache. That's your colon. Clear? So this is your lecithin. Fine, this is your lecithin. So its structure is given in NCERT. So obviously you need to remember this. Okay. Done? Fine. So most common type of phospholipid it is. So we have another phospholipids also, like we have phosphatidic acid also. So ultimately, see, let me relate it with the plasma membrane. So you know that the plasma membrane is made up of these phospholipids. Okay, plasma membrane, it is made up of these phospholipids. So when you talk about this head region, you know that this head is what? It is hydrophilic. This head is what? It is hydrophilic. And when you talk about this tail part, this tail is what? It is the hydrophobic. Okay, so that is why it is not facing the surrounding. Clear, bache? So head is, you know, that polar, and when you talk about the tail is your non-polar. Okay, the tail is your non-polar. And here, when you talk about this tail, the tail is going to have saturated and unsaturated fatty acids. It's not mandatory that totally it is made up of unsaturations. No, unsaturated and saturated fatty acids are there. So wherever there is a bend, there is a kink, it is showing the unsaturation. And here in the head region, you will see your glycerol, you will see your phosphate here, right? So you are going to see all the groups here, bache, basically. Done? So your phosphate, your glycerol part. Here you will be having your phosphate and your glycerol part, your polar part. Clear? Clear, bache? Sure? So nitrogenous compound in the lecithin is colon. This is what you need to mention. So one is phosphatidic acid. I told you already about this. That's all. So this is what you need to remember. Okay. Okay. So one more is there, that is your sphingolipids. Sphingolipids. So these sphingolipids are also bache, a type of conjugated base, but here at place of glycerol, right, glycerol, we'll be having the amino alcohol is there, right? Amino alcohol is present. What is present here? Amino alcohol is present here. Clear bache? Clear? And that amino alcohol is sphingosine. Sphingosine. So you will find the sphingolipid in the nerve cell. Basically, myelin sheath is formed with it. Fine. It forms myelin sheath. And glycolipids are also there. So obviously the carbohydrate part is attached to the lipids. Okay. The carbohydrate part is attached to the lipids. So if there is any doubt, please let me know. Sure. Please let me know. Sure, bache? Done? Okay. So here you people can see the cholesterol. Bache, it is the example of a derived lipid. And it is present in our plasma membrane of animals as well. Right? It is playing role in maintaining that fluidity. Okay. Right. So now cholesterol is the example of, as I said, it is the example of your derived lipid. Now, what is the meaning of derived lipids? What is the meaning of derived lipid? So, as I said in the case of derived protein, so from which is simple lipids or conjugate lipids, we are going to get them. Right? These derived lipids, we will obtain them from the simple lipids and the conjugate lipids. And here you will see the structure will be a bit complex. Okay? The structure is going to be complex here. Like you will see rings and all. What are you going to see here? You people will see some rings here. Okay. So when you talk about the derived lipids, the most common example is your steroids. Right. And in these steroids, you have the example of sterols. You have the example of sterones. Okay. Look at the functional group. Sterols, all, alcohol, on, ketone group. Ketone group is there. Right. So steroids are the most common type. You know that steroids are your derived lipids. So they have the tetracyclic structure. Right. What do they have? These steroids are having the tetracyclic structure. So they have two forms, bache, the sterols and the sterones. So the cholesterol, that the word that we use, cholesterol. Cholesterol is a type of steroid. Clear? It is the type of steroid. Right? So when bache I used to, when I was teaching you plasma membrane, I clearly mentioned that in the plasma membrane of animal cell, okay, in the plasma membrane of what? Plasma membrane of animal cell, what is present? Cholesterol is present. Which sterol is present? It is the cholesterol which is present. Right? So, but in the case of plant cells, you do not have the cholesterol. Some other sterol is there. Okay, in the plant cell membrane, cholesterol is absent, some other sterol is there. Are you getting it? Cholesterol is absent, some other sterol is there. Done, bache? So this is about the derived lipids. Fine, this is about the derived lipids. Clear? So it is the cholesterol which is most abundant, of course. We know that. So if I talk about the functions of, important functions of cholesterol or steroids. So see, you know that steroids are also used for making the sex hormones, right? We know, we used to say, na, there are some sex hormones as the hormones, they are having different nature. They can be made up of amino acid, they can be made up of proteins, right? Lipids are also there. So steroidal hormones are also there. Okay, steroidal hormones are also there. So that steroidal hormones will include your sex hormones, all the sex hormones. Fine, all the androgens like testosterone, estrogen, progesterone, for their synthesis, cholesterol is required. Okay, cholesterol is required. And do you remember the function of smooth endoplasmic reticulum? So basically in smooth endoplasmic reticulum, this cholesterol synthesis or this lipid synthesis will take place. Okay, this cholesterol synthesis or this lipid synthesis will take place. Fine, bache. So steroidal hormones, sex hormones are steroidal hormones. Hormones of adrenal cortex, they are also steroidal in nature. है ना? They are also steroidal in nature. Clear? Fine. Is it clear? Right? Examples are important here. Now I hope you know about the ecdysone. I hope you know about the ecdysone. Ecdysone. In the case of insects, when you talk about the insects, there is metamorphosis, there is molting. In the case of insects, there is metamorphosis, there is molting, yes or no? Yes or no? So during the molting, for ecdysis, in the insects, we see this ecdysone. So this ecdysone is also a kind of steroid, but it is a sterone. Okay, it is a sterone. Clear, bache? Yes, is that clear? So it is ketonic steroid. Okay, it is ketonic steroid. Fine. Sure. So yes, adrenal cortex, adrenal cortex, steroidal hormones, but these adrenocorticoids, if I'll be more specific, so these adrenocorticoids, they are sterones basically. Okay, so that's it. This is all about the derived lipids that you need to know. So just look at the diagram once again. So you see 4 rings are there, na? Tetracyclic it is. Looks like a chair and all. Right? So it is considered as most decorative biomicromolecule as well. Isn't it? It is considered as most decorative biomicromolecule as well. Okay? So this is all about the lipids. Let's revise it from NCERT and then we are going to start the next part, which is your nucleic acid. Okay? Which is your nucleic acid. Now to understand the nucleic acid, we should know about the sugars. You already know about the sugar. I'll tell you about the nucleonitrogenous bases and then that topic will also be easy. So that topic is very important for your molecular basis of inheritance. Okay. So try to make the notes and please revise it. Please revise it properly. Okay. So lipids, you know that they are generally water insoluble, but they are soluble in the organic solvents like chloroform, like benzene. Fine. So they could be simple fatty acid. A fatty acid is having a carboxylic group attached to the R group. So this R group could be, now you can read it here, methyl or ethyl or higher number of CH2 groups. Isn't it? Isn't it? It can be methyl, it can be ethyl, or it can be higher number of CH2 groups. So 1 carbon to 19 carbons. Right? So many times students, they get confused here that ma'am in NCERT it is given as 1 to 19 carbon. See, number of CH2 group is 1 to 19. Like if I talk about the arachidonic acid, obviously number of CH2 group is going to be 19, but ultimately total, right, you have to include the COOH as well. So ultimately the carbon number is 20 there, na? Ultimately the carbon number is 20 there, na? Clear? So palmitic acid has 16 carbons, I told you. Arachidonic acid is having 20 carbon, including the carboxyl carbon. Okay, including the carboxyl carbon. Done, bache. So fatty acid could be saturated without double bond, unsaturated with 1 or more carbon-carbon double bonds. Okay, so another simple lipid is glycerol. So glycerol is also considered as simple lipid. This is the MCQ and it is your trihydroxypropane. Clear? This is your trihydroxypropane. Is that clear? It is your trihydroxypropane. Done, bache. So many lipids have both glycerol and fatty acids. So they are found esterified with glycerol. They can be mono, dry and tri. So during the fat digestion, you know that first of all emulsification of fat will be there. The large globules, we are going to break it into small globules. Then further the triglyceride, diglycerides, the monoglycerides will form, and finally we will separate fatty acids and glycerols as well. Okay, so in the digestion also this part is very important. So try to understand this. But biochemistry is very interesting. Right, just listen to me carefully, revise it from NCERT, then everything will be crystal clear. And here in our marathon sessions, right, you guys can see that we are even including the NCERT and we are discussing that in detail. Right, we are discussing the topics, the points in detail. Okay. Okay, so these lipids are fats, the oils based on the melting point. So this line is important. Your oil, they are having low melting point, right? right? So they remain oil in winters because their melting point is low, right? Done, bache. So some lipids have phosphorus and phosphorylated organic compound, told you already. Just phosphatidic acid is just having this phosphorus. Phosphorylated organic compound, your lecithin. Okay, so these are phospholipids. They are found in cell membrane. Lecithin is one example. And some tissues, especially the neural tissue, have lipids with more complex structure. As I said, sphingolipids are there. They are basically covering the myelin sheath, so the part of nervous tissue it is. Nervous system it is. So living organisms, you know that they have a number of carbon compound in which heterocyclic rings are there. So now next part is from the nitrogenous bases. This we will discuss after 1 minute break. So tell me if there is any doubt, just ask it from me. Okay, just ask the doubt and then we'll start the next part, that is your nitrogenous bases. Hanji, any doubt? Sure? Bache, different reasons can be there, Shiva Kumar. Sometimes, you know, some painkillers, they include the steroids. Sometimes some obviously hormonal medicines are also having steroidal preparation, right? So many things are there, right? It depends upon the disease. Anything else? So can we start? Can we start? Bache, I love chai, tea. I don't like coffee much. Like rarely I used to have coffee. Any other question or can we start? The next part is very interesting. The next part is very interesting. Okay. So next is nucleic acid. Okay. So nucleic acid is again the biomacromolecule, isn't it? It is also present in retentate. It is also present in acid insoluble fraction, right? What are we discussing? We are talking about the nucleic acid, and nucleic acid is also there in acid insoluble pool. Okay, it is the part of retentate. Done, bache. So when you talk about nucleic acids, you discuss DNA and RNA. Okay, DNA and RNA. So DNA is your, yes, DNA is your deoxyribonucleic acid, isn't it? DNA is your deoxyribonucleic acid. Clear? And when it comes to the RNA, it is your ribonucleic acid, right? It is your ribonucleic acid. Now, bache, when we discuss the nucleic acid, in the nucleic acid, right, in the nucleic acids are basically the polymers of nucleotides. Okay. So they are the polymers of nucleotides. Fine. Fine. What are they? They are the polymers of nucleotides. So when you discuss the nucleic acids, so you say that they are the polynucleotide chains, right? What are they? They are the polynucleotide chains. Like many nucleotides, they are going to join together and they will form the chains. Fine. So nucleic acid, what is it? It is a polymer. It is made up of nucleotide chain. So that is why we are also using the word polynucleotide chain. Clear, bache? That is why we are also using the word polynucleotide chain. Now, this is how you guys can imagine. One nucleotide will join with another nucleotide, will join with another nucleotide. Fine. So this these they will form a chain, and that chain is going to form the DNA and RNA. Okay, that chain is going to form the DNA and RNA. Done? Done? So if you want to understand the nucleic acid, firstly you should know the nucleotides, and if you want to understand the nucleotides, you should know about the nitrogenous bases. Then you need to know about the sugars. Then you need to know about the phosphoric acid. Clear, bache? Clear? So one by one we need to understand it. Okay? So this part is easy but little tricky. Okay, so please try to focus. Try to focus. So nucleotide. So if you want to understand it, you should know about the nitrogenous bases. You need to know about the sugars. You need to know about the phosphoric acid. Clear? Clear? So let's start with the nitrogenous bases. So students, if we are talking about the nitrogenous base, so one thing is obvious that these are the compounds having nitrogen in it. Obviously, nitrogenous base means compounds having nitrogen in it. Now here in nitrogenous bases, we talk about the purines, right? And we talk about the pyrimidines. What are we going to discuss here? We are going to talk about the purines. We are going to talk about the pyrimidines here. I'm repeating it again. We are going to talk about the purines. We are going to talk about the pyrimidines here. Purines, pyrimidines, purines, pyrimidines, purines, pyrimidines. This is very important. And bache, for understanding the DNA structure, you should know about these things. Okay, we need this part in molecular basis of inheritance as well. So please try to focus. Okay, please try to focus. So what do we have here? We have the purines, pyrimidines, purines, pyrimidines. Now, so we have a trick here. For purines, it's puga. And for pyrimidine, the trick is your cut. Fine. For purines, the trick is puga. And for pyrimidines, the trick is cut. So when it is puga, purine, Pu stands for purine, purine. G stands for guanine. A stands for adenine. G stands for guanine. A stands for adenine. Clear, bache? G stands for guanine, A stands for adenine. Now when you talk about the pyrimidines here, C stands for cytosine, U stands for uracil, T stands for thymine. Okay, T stands for thymine. Are you getting it? So we have 5 nitrogenous bases, 2 purines, 3 pyrimidines, right? 5 nitrogenous bases are there, 2 purines, 3 pyrimidines. So purines are your adenine and guanine. Adenine denoted by A, guanine denoted by G. Pyrimidine, cut, cytosine, uracil, thymine. Cytosine denoted by C, uracil denoted by U, thymine denoted by T. Okay, okay. So now when you're talking about the purine, just look at the spellings here. 9, 9. Okay, 9, 9. So basically it's a 9-membered ring. What will you see here? 9-membered ring. So purines, they are 9-membered ring. Okay, and when you talk about the pyrimidines, it is a 6-membered ring. Okay, it's a 6-membered ring. Fine. So now in the case of purines, your nitrogen is also there. So you will see nitrogen at, right? 4 nitrogens are present in the ring, basically. 4 nitrogen are present in ring, right? Ring is 9-membered. Okay, 4 nitrogen are present in ring and they are present at position number 1379. 1379. That's important. At 1379. 1379. And here it is a 6-membered ring having 2 nitrogen atoms Okay, at position 1 and 3. Clear? At 1 and 3. Are you getting it? At 1 and 3. So, bache, this information is important. It's literally important. Clear? It's literally important. So, please note down. Do it quickly. Yes, do it quickly. Then we'll start the next part, the structures and all. ठीक है 1379 1379 Wow, for the first time, right? Without any mistake, I made it. Purines. They are purines. What are they, bache? They are purines. So 1, 3, 7 and 9. 1, 3, 7 and 9. Okay. So it is at 1, 3, 7 and 9. Clear, bache? Clear? So 1, 3, 7, 9 it is. 1, 3, 7, 9 it is. Okay. So alternate double bonds are also there. So this is 1, 2, 3, 4, 5, 6, 7, 8 and 9. So these are the purine. So you can say it is a, you can see here it is a 9-membered ring. Total 4 nitrogen are present in the rings. And now when it is pyrimidine, so of course the structure is like this. Isn't it? The structure is like this. Clear, bache? So 1 and the third one here. Clear? Is that clear? And of course the alternate double bond and single bond is also there. Fine. The alternate double and single bond is also there. So you guys can draw it. So 1, 2, 3. Fine. And here also in the pyrimidines also you will see the double bond. So here and okay. Done. So purines and pyrimidines. Purines and pyrimidines. Now let's just look at the structure of purines. Can I say that in the structure of purines you are having one pyrimidine ring also? It is just like a pyrimidine ring, isn't it? Isn't it? This part is just a pyrimidine ring. Yes or no? Yes or no? It is having 2 nitrogen. It is having 1 to 3 carbon, 2 nitrogen. Don't you think that it is a pyrimidine ring? Having 2 nitrogen and 3 carbon. Having 2 nitrogen and 4 carbon, sorry. है ना? And this one is the imidazole ring. Okay, this one is the imidazole ring. So when you talk about the purines, so purines are having one pyrimidine ring plus one imidazole ring. One pyrimidine ring and one imidazole ring. So pyrimidine mein you can see that we have 2 nitrogen and we have 4 carbon. And here you people can see 2 nitrogen and 3 carbon. Okay, 2 nitrogen and 4 carbon and 2 nitrogen and 3 carbon here. Done? So this is purine, pyrimidine. Purine, pyrimidine. So purines mein, I am repeating this again, the trick is puga, and pyrimidine mein it is cut, that is CUT. That is cytosine, uracil and thymine. Now, what is the point that you need to take care here? Right. See, your cytosine and your thymine. These are 2 pyrimidines which are present in DNA. Okay. Cytosine and thymine. See, Y is common. Y and Y is common. So, these are the pyrimidines present in DNA. Right. These are the pyrimidines present in DNA, right? These are the pyrimidines present in DNA. And when you talk about the uracil and the cytosine, see, uracil, R is there. So obviously it is the part of RNA. So that is a difference in between DNA and RNA. Okay, that is a difference in between DNA and RNA. Clear, bache? So puga and here pyrimidine, right? So cut is the trick. So this is what you need to remember, and that's important. Fine, that's important. So nitrogenous bases means nitrogen containing compound. Now let me check the structures. See, okay, see here the structures. Pyrimidine 1, 3 and alternate double bonds are there, 1, 2, 3. Clear, bache? And purines mein 1, 2, 3, 7, 9 and this one is the 8. Fine, bache? So now cytosine, thymine, uracil. So how can you differentiate it? Many times students, they used to ask me, ma'am, do we need to remember the structure? Yes. In the case of nitrogenous bases, yes, you have to remember the structure, bache. You should remember it. It is important. Now see, one thing is very common, na, bache. If here you have the N, right? Here you have the N, it is attached to the hydrogen here, right? Here, because of the presence of double bond, the valency is satisfied. So when it is cytosine, you will see C double bond E, right? The carbon present in ring is forming a double bond with the oxygen, and here one carbon is attached to this NH2 group, right? Try to remember it. Now, just compare thymine and uracil. You know, thymine is also known as 5-methyl uracil. It's a very common question. Okay, thymine is also known as 5-methyl uracil. The thymine, it is also known as 5-methyl uracil. See here, to the 5th carbon, this one is 1st, 2nd, 3rd, 4th and 5th and 6th. Yes, 1, no. Okay, 1, 2, 3, 4, 5 and 6. So 5th carbon of the ring is having the methyl group here. And here in the uracil, this methyl group is absent. This is the only difference present here. Clear, bache? This is the only difference present here. Done, bache? Done. So it is the thymine, it is the uracil. Thymine is also known as 5-methyl uracil. It is, you can say that uracil is more reactive, right? See, when we talk about the DNA and RNA, you know that DNA is the genetic material. RNA is the genetic material, but in the case of viruses. Otherwise, RNA is doing other jobs like messenger RNA. It is helping in protein synthesis, helping in the formation of your ribosomes as well. Isn't it? This is what we already know. Now, when you talk about this RNA is also the single-stranded molecule, right? And when you talk about the DNA is double-stranded. It is believed that with time, during evolution, from RNA, DNA evolved, right? And it was important as well. Why? Because the genetic material should be less reactive. It should not be more reactive, right? It should be stable. It should allow the mutations, but at a very slow rate. Okay, at a very slow rate. Okay, so here in the case of uracil, in the case of RNA, it is having uracil, it is having ribose sugar, which is making it very, more reactive, and moreover it is single strand. But in DNA, such things are not there. Okay, so it is from evolutionary point of view, it is very important. So when you will do that now, that the comparison in between DNA and RNA, you will understand it. Yes, Krishna, it's very important for class 12. Okay. So next is what? Next is purine. So see, adenine, guanine. This is how we pronounce it. Guanine. O is there. So see. So if I'll get the structure in the paper, if I'll get two structures in the paper, right, by looking at the structure, I can conclude one thing, that it is a 9-membered ring having one pyrimidine ring and one imidazole ring, right? This is what we all can see and can conclude. Now, if in that ring, if C double bond O is present, then obviously it is guanine. It is guanine. Are you getting it? It is guanine. Are you getting it? And now here in the adenine, you can see only amino group is present here, right? It is attached here. That's all. Done, bache? That's all. So in guanine, one amino group is there and one, the C double bond O group is also there. Okay, bache? So this is about the, yes, what is it? It is about the nitrogenous bases. Now the next part that you need to understand is, sugars. Okay, so sugars, they are the carbohydrates, right? So here we will talk about the monosaccharides. Okay, we are going to talk about the monosaccharide. Which monosaccharide? The pentose. Okay, the pentose, the aldose pentose. The aldose pentose means the functional group is aldehyde and 5 carbons are there. Okay, so the ring structure, I hope you remember, it is a furanose, isn't it? It's a furanose, isn't it, bache? So ribose is what? It's a pentose sugar. So let's say this one is ribose and this one is deoxyribose. Okay, this one is ribose, this one is deoxyribose. So both of them are monosaccharides. Okay, so ribose is having the it is having the formula C5. Do you remember that general formula CnH2nOn? So here the carbon number is 5. So the formula will be C5H10O5. And when you talk about the deoxyribose, it is an exception. It doesn't follow the general formula. Fine. It doesn't follow the general formula for monosaccharides, isn't it? It doesn't follow the general formula for monosaccharides. Clear, bache? Clear? So here the formula is C5H10O4. C5H10O4. So this is the first carbon, this one is the second, third, fourth and fifth oxygen. First carbon, second, third, fourth and the fifth carbon, right? So fifth, and this is the fifth oxygen. Now see, see deoxyribose and ribose, see everything is same, bache. OH, obviously OH is above, right? H is below, so it is the beta deoxyribose sugar. It is beta. Okay, so alpha means hydrogen above, beta means hydrogen below, right? Do check my last part. Okay, so here also, but when you look at the second carbon, see in the case of ribose sugar we have the OH also, right? And here third carbon is again HOH and CH2OH and the H is there, right? But now when you compare the ribose sugar with the deoxyribose, deoxy without oxygen, right? Deoxy without oxygen, and this oxygen is absent at the carbon number, or you can say that on the carbon number 2. So this is what you need to remember. This is a very important point. Okay, this is a very important point. Clear, bache? Clear? So let's draw it. So it's OH, H, it's OH, H. So here it will be H and H, again H, OH. So here it will be CH2OH and H, it's ribose. Fine. So that's right. It's ribose, it's deoxyribose. So always remember it is the second carbon where the oxygen is absent in deoxyribose. So that is why the general formula is C5H10O4. Okay, it is C5H10O4. Clear, bache? It is C5H10O4, right? So next is your phosphate group. Next is what? Next is your phosphate group. Okay, it is your phosphate group. So P double bond O, OH, OH and OH. So next is what, bache? Next is the, yes, next is the phosphate. Okay, or you can say that phosphoric acid it is. It is not phosphorus, it is phosphoric. Okay, it is a phosphoric acid. It is not phosphorus acid, it is phosphoric acid, which will be used here. So obviously it is negatively charged, and that is why the nucleic acids, they are negatively charged. Okay, this phosphorus is adding the negative charge there. Now I told you, if you want to understand the if you want to understand the nucleotide, so you should know about what? You should know about the nitrogenous base. Now you know about the nitrogenous base. Now you know about the sugar. So now what you need to understand? You need to understand the nucleosides. What you need to understand, bache? You need to understand the nucleoside. What is it? Nucleoside. See, N stands for nitrogenous base. S stands for sugar. So when your nitrogenous base, it joins with sugar, what are you going to get? You will get a nucleoside. So now the point here is how these nitrogenous base and sugar will join. Of course, a bond will be there. Which bond? It is N-glycosidic bond. It is N-glycosidic bond. Clear? What is it? It is N-glycosidic bond. Okay? Okay? Done, bache? It is N-glycosidic bond. So, let's take the example here. Right? So, I'm not going to consider any, I'm not going to take the example of any particular amino acid. Let me tell you. 1, 2, and 3. Okay. Again, I'm not going to take the example of any particular nitrogenous base here. Let's say this is one nitrogenous base. Obviously, it is pyrimidine. Another is sugar. Right? The another one is sugar. 1, 1 or 1, 9. Oh, please observe it very carefully. So it's a ribose sugar. Okay. So this is the carbon number 1. It's the ribose sugar. This is the carbon number 1. This is the nitrogen number 1. So here in between these two, what is going to happen, bache? Here in between these two, the glycosidic bond will form. What is going to happen here? The glycosidic bond will form in between these two. Okay. The glycosidic bond is going to form in between these two. So you can say the first carbon of your sugar and the first nitrogen here. Okay, the first nitrogen here. Are you getting it? Yes, are you getting it? Let's say it is like this. Okay, okay. So in the nucleoside, there will be a bond formation in between your nitrogenous base and the sugar, and that bond is known as N-glycosidic. Why N-glycosidic? Because one carbon is there, no doubt, but another is the nitrogen. Another is the nitrogen. So that is why we use the word N-glycosidic bond. Okay, N-glycosidic bond. So, bache, if it is a pyrimidine, then bond will be there in the first carbon of sugar and first nitrogen of pyrimidine. Okay, and if it is a purine, then bond will form in the first carbon of sugar, in between the first carbon of sugar and the 9th, right, nitrogen, like basically the nitrogen which is present on the 9th position. Clear, bache? Done? So, done? Okay. So, this is a nucleoside. What is it? It is a nucleoside. Now, see, if adenine is the nitrogenous base. If adenine is going to form a bond with the ribose, now you will call it as adenosine. Right? If the sugar is ribose, it will be adenosine. If adenine is going to form the bond with deoxyribose, okay, if it is going to form the bond with deoxyribose, so it will be deoxyadenosine. So in the paper, they can ask you what is the difference in between the adenine and adenosine. Adenine is a nitrogenous base, adenosine is a nucleoside. Adenine is a nitrogenous base, adenosine is a nucleoside. Clear? Clear? Right? So guanine plus ribose, guanosine. Guanine plus deoxyribose, deoxyguanosine. Cytosine plus ribose, cytidine. Cytosine plus deoxyribose, deoxycytidine. Thymine plus deoxyribose, because thymine is absent in RNA. So it is always thymine plus deoxyribose, deoxythymidine. Uracil plus ribose, it will be uridine. Clear? So they are nucleosides. What are they, Soumyadeep? They are nucleoside. Clear, bache? Clear, bache? So next is your nucleotide. From this T, you have to remember that it's 3. Okay? Tide, T, 3 it is. So there are 3 things basically. So nitrogenous base, sugar plus phosphate. Nitrogenous base plus sugar plus phosphate. So bache, nitrogenous base and sugar, they are connected together by N-glycosidic bond. Okay, and when you talk about this sugar and phosphate, here you people will see phosphoester bond, right? You will see phosphoester bond. Clear bache? What will you see here? You will see phosphoester bond, nucleotide. So let's say if it is adenine plus ribose plus phosphorus, so it will become adenosine, right? Adenine plus ribose is already adenosine. So adenosine, only one phosphate is attached there. So adenosine monophosphate. It's going to be adenosine monophosphate, and you can also call it as adenylic acid. It is also the adenylic acid. Is that clear? It is the adenylic acid. So adenosine monophosphate and adenylic acid is same. Okay, it is a nucleotide. It is a nucleotide. So obviously sugar, this is how I remember. Sugar is sweet, right? So even nitrogenous base wants to form the bond with sugar, even the phosphorus. Clear, bache? Done? It is phosphoester. So bache, when it is a single, okay, nucleotide, the bond is phosphoester. It is not phosphodiester. It is phosphoester. Clear? It is not phosphodiester. It is the phosphoester bond. Is that clear? It is the phosphoester bond. Done, bacho? Yes? Yes or no? Okay, so let me show you the bond formation here. So we have the sugar, HOH, wait, so it will be OHH, HOH, HOH, HCH2OH. So now here you have this P double bond O, OH, OH and OH. So from this part, the water will go out, right? The water will go out. There will be the release of water. So obviously, what are you going to get here? Tell me, what are you going to get here? Yes or no? Right, because it was CH2OH now. Let me write down. See, it was CH2OH now. CH2OH. So the water will go out. Right, this water will go out. Minus H2O will be there. So you are left with CH2O, P double bond O, right, and this OH here. Clear, bache? Right, so this is your phosphoester bond. Okay, now it is not C double bond O and C. It is your P double bond O and C. Fine. Done. So it is phosphoester bond. It is phosphoester bond. So the fifth carbon of the sugar, right? The CH2, the OH group of this fifth carbon of the sugar, it will participate in the bond formation with this phosphorus. Okay, so it is a nucleotide. So this is your homework. You will write a short note on all these, that write down the name of nucleic nitrogenous bases, then nucleosides, then nucleotide. Okay, then nucleotide. So all these nucleotides, they are going to join and they will form. See, sugar, first of all, understand it. Nitrogenous base, phosphorus. Sugar, nitrogenous base, phosphorus. Sugar, nitrogenous base, phosphorus. Clear, bache? So now the sugar, the 3-OH, the third OH will take part in the bond formation here. So in a polynucleotide chain, okay, in a polynucleotide chain, when it is a polynucleotide chain, when many nucleotides they join together, then here in polynucleotide chain, you will see phosphodiester bond. Okay, which bond will be there, bache? Phosphodiester bond. Fine, phosphodiester diester bond. Okay, done? Clear? Sure? Sure. So, bache, in detail, we will discuss it in next class. So, there is only one part of this chapter left where you are, so where we will discuss the structure of DNA, but that is not so important because that we will cover in your, what we call it, in molecular basis of inheritance. So, see, so we will talk about this metabolic basis for living. It's very interesting. Okay, the living state and the enzymes and the chemical reactions. So we just need one more part and we will cover it. Okay, so now you know what you have to do. Please put some good comments, right? If you like the chapter, obviously, if you have attended the session, right, it means you like my teaching. So please put some good comments in the comment section and be the part of NEET Energize batch. If you want to join it, please don't waste your time. Okay, from 1st of August, I will start plant physiology. And moreover, right, if because of any reason you cannot join the NEET Energize batch, please join the All India mock test series and start giving the mock test from now itself. Okay, so thank you so much for your time. Do revise it from NCERT. I'll see you in the next